We request you to take your seats and be prepared as we Once again, Ayubovan, Namaste, and a very good afternoon to all of you. And we warmly welcome you to Rasashastra Sambhasha 2023, the 6th edition of Sri Dhootapapeshwar Rasashastra Sambhasha 2023. As you know, this unique Indo-Sri Lankan platform of knowledge sharing was happening for nearly 6 times, this being the 6th edition of it, bringing you a history of 151 years of Sri Dhootapapeshwar Limited, bringing you the best of Rasashastra all the way from India. Alpamatropayogiitvat daruchera prasannata shipramarogyadaitvat maushadebhyo diko rasa. As you all know, that we can prescribe it in tiny doses. It will not cause any unpleasant feeling to the patient and will give the healthy action so quickly than any other drugs. Hence, Rasa Aushadha is greater than any. So that's why we are here today. All of you are here today at the Rasashastra Sambhasha, the 6th edition of it. We once again warmly welcome you all as we are about to commence this evening of knowledge sharing. We've liked you to request to stand in attention for the national anthems of Sri Lanka, followed by the national anthem of India.
Sri Lanka, Namo 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 Jai.
Please be seated. We shall now have the lighting of the traditional oil lamp, and I shall invite the following dignitaries to do the honors. Rasashastra Sambhasha, Hayvana Vatavatat, ඔබ වෙත දිගහැරීමට මෙවරත් ශ්‍රී දූතපාපේශ්වර ලිමිටඩ් සූදානම්. ඉතින් ඒ සඳහා පැමිණි ඔබ සැම පැමිණෙමින් සිටින ඔබ සැමවත් අපි තාම ගෞරවයෙන් ආදරයෙන් පිළිගනු ලබනවා. And also I would like to mention that we are live streaming it on the Facebook page of Sri Dhootapapeshwar Limited. We warmly welcome all who join from India as well, who are joining us online. I invite the following dignitaries to light the traditional oil lamp, starting with our chair of the forum, Professor S.G. Ranasinghe. We also invite the Commissioner of Ayurveda, Dr. Dhammika Begunawardena, also Dr. P. Hewagamage, the Technical Commissioner of Ayurveda. We also invite the speakers who are present with us today to light the traditional oil lamp, Dr. Yogesh A. Kulkarni, Professor Priyani Paranagama, Professor S.K.M.K. Harapadeniya, Dr. Subhamangalacharya, Dr. Priyadarshini Gunaratna, and Dr. Mukesh Chowda. We also invite the co-chairs of today's forum, Dr. S.L. Vevalvala and Dr. K.P.K.R. Karunagoda. Not forgetting the Chairman and Managing Director of Sri Dhootapapeshwar Limited, Mr. Ranjit Puranik. Thank you for joining the lighting of the traditional oil lamp. And we shall now remain standing for the Dhanvantari Puja and invoke blessings on the gathering. I'm sorry, you can be seated as we're going to start the sessions along with inviting our dignitaries to the head table. I would like to start with inviting the chairman of today's forum, Professor S.G. Ranasinghe, who is a well-respected and admired figure in the Ayurveda fraternity of Sri Lanka. He holds MD and PhD in Ayurveda and has held key positions of Director, Institute of Indigenous Medicine, and Senior Consultant to the Ayurveda Teaching Hospital, Borella. He has to his credit the stupendous achievements in his long-spanning career as clinician and academician. And I would like to invite the co-chairs of today's forums. The forum would have two eminent academicians as co-chairs, Dr. S.L. Vevalvala, Senior Lecturer and Head of Department of Ayurveda Basic Principles at Faculty of Indigenous Medicine, Gampaha Wickramaratche University of Indigenous Medicine, and Dr. K.P. K.R. Karunagoda, Senior Lecturer and Head of Department Shalishalakya Prasuti Tantra Kaumara Brutya at Faculty of Indigenous Medicine, University of Colombo. And now I would like to invite Dr. Ketan Bhabkar, Manager Exports, Sri Dhootapapeshwar Limited, to address the gathering and to officially welcome you all.
Ayubovan, welcome, namaste. I welcome you all to Rasashastra Sambhasha 2023. In 2015, we envisioned a knowledge-sharing platform and launched this event. Ever since its launch, the contributions from the stalwart speakers, support from India and Sri Lankan Ayurveda fraternity has enhanced its grandeur to this 6th edition of Rasashastra Sambhasha. In this 6th edition of Rasashastra Sambhasha also, we have highly accomplished dais and speakers who will be sharing their opinions, their research work, their experiences in using Rasaushadhis safely and effectively in their treatment of Ayurveda physician to attain the ultimate aim of Ayurveda Chikitsa, that is Rugnahita, or the well-being of the patient. So this will be an opportunity for us to gain the insights into different facets of this unique science of Rasashastra and Ayurveda from this forum. So I will not be barrier in between us all and the knowledge of Rasashastra and Ayurveda. I welcome you all again, and with the permission of Honorable Chair and co-chairs, we can start the deliberations of this scientific session. I welcome you all. Thank you.
Thank you, Dr. Ketan. And let us start the first session of today's scientific forum. I invite Dr. Yogesh A. Kulkarni, who is an Associate Professor and Associate Dean at Shobhaban Pratapbhai Patel School of Pharmacy and Technology Management, SPKMs and NMIMs, Mumbai, India. He is an eminent academician and researcher. Over past 18 years, he has conducted numerous research projects in various fields, including herbal drugs, natural products, diabetes, diabetic complications, and neurodegenerative diseases. He has 110 publications in peer-reviewed journals to his credit and has authored many books. Dr. Kulkarni is recipient of prestigious DST Inspire Faculty Award, awarded by Government of India. He has been featured in top 2% scientists in the world, list published by the Stanford University and elsewhere in 2021 and 2022. Dr. Yogesh Kulkarni would share his interesting insights on role of Rasaushadhi in diabetic complications and evidence-based revalidation. Over to you, sir. And we also invite Dr. Mukesh Chowda to join the head table.
dignitaries on the dais, off the dais, doctors, healthcare professionals, students, ladies and gentlemen. Good afternoon to everyone. So today I'll be talking on a topic exploring potential of Vasanta Kusumakar Rasa in diabetic complications, molecular pharmacology approach. So diabetes, everyone knows it is nothing but what? It's a chronic disease which occurs when the pancreas is no longer able to make insulin due to some defects in pancreatic beta cells or destruction of pancreatic beta cells. Not being able to produce insulin or use it effectively leads to increased glucose levels in blood, which we call it as hyperglycemia. And long-term high glucose levels, persistent hyperglycemia, is associated with different types of complication. We call them as diabetic complication. And everyone knows the prevalence of diabetes is continuously increasing, and we can say that in 2050, around 60 to 70% people, they'll be suffering from diabetes. Now coming to diabetic complications, this persistent hyperglycemia damages important organs in our body if it is not well managed. Like it can affect eye, it can affect heart, it can affect kidney, neurons, brain, etc. So based on these damages, different types of organs which are being damaged, these complications can be categorized into microvascular and macrovascular complication. So when it is a small blood vessel which is responsible for damage to any specific organ, we call it as microvascular diabetic complication. And when large blood vessels are involved, we call it as macrovascular complications. So it leads to diabetic retinopathy, diabetic nephropathy, diabetic neuropathy, cardiomyopathy, encephalopathy, etc. So this persistent hyperglycemia management is very important aspect when we want to treat or manage diabetic condition. Now when we talk about diabetic complication, these diabetic complication, as their name suggests, they are too complicated when it comes to detailed pathophysiology. There are many factors like metabolic factors, uncontrolled glycemia, uncontrolled lipids, they are linked with each other. Different types of hemodynamic parameters, we generally see that, diabetic people, they suffer from different types of cardiac disorders. Blood pressure, salt fluid balance, and of course, the last one is the genetic factors, which can also play important role in development of these diabetic complications. Now, all these factors, when we combine together, metabolic, hemodynamic, and even genetic, it leads to different types of changes at cellular level. And these cellular changes, it may lead to modifications in gene or modulation of different types of proteins involved in progression of these diabetic complications. At the end, the immune cell recruitment at various organs leads to severe damage, causing cell dysfunction and cell death. This is applicable to all organs which face this persistent hyperglycemia. Vasanta Kusumakar Rasa, it's a well-known medicine in Ayurveda. It's a classical Ayurvedic formulation indicated in Prameha, and it's a unique combination of minerals and herbs that provides natural immunity to body cells and help in the treatment of weakness which is caused in diabetic condition. Rasashastra, they work on different slotas and penetrate into deepest tissues carrying highly potent charge. And of course, we know that VKR is an important formulation which is manufactured and marketed by various herbal drug companies. Now coming to this composition, I hope that everyone knows composition, but it consists of Suvarna Bhasma, Rajat Bhasma, Vang Bhasma, Naga Bhasma, and some herbs. And each of these components has been scientifically studied and scientifically reported to have anti-aging property, immunomodulator property, then cardiotonic property. Some constituents have also been reported to have excellent effect in obesity. So indirect scientific evidence of all these conditions suggests that VKR will be useful in diabetes-related complications. There are many papers published on this excellent formulation. Few papers we have came across that it is useful in management of retinopathy, type 1 retinopathy. Everyone knows that type 1 and type 2 diabetes, the complications, they may differ when it comes to pathological conditions. So this Vasanta Kusmakar Rasa has been reported to have effect in diabetic retinopathy. But as we know that 90% diabetic patients, they suffer from type 2 diabetes. Type 1 diabetes, of course, the patients, the number is considerable, but the majority diabetic patients, they suffer from type 2 diabetes. So we thought that this particular known formulation, it must be tested in type 2 diabetes related complications. So coming to this hypothesis or rationale for the present study, we know that unmet need related with treating diabetes and diabetic complication. There are no specific drugs approved by any regulatory agencies for managing, or we can say preventing, these diabetic complications which affect majority of diabetic people. Traditional classical formulation of Ayurveda focuses on holistic approach towards the treatment, and the main advantage of this is potency, effectiveness, and longevity. So that's why we proposed to study this particular formulation in animal models of diabetic complications. So these are the objectives. We studied this Vasanta Kusmakar Ras in type 2 diabetic nephropathy, type 2 diabetic neuropathy, cardiomyopathy, and type 2 diabetic retinopathy. Coming to the first complication, neuropathy, the increased prevalence of diabetes has inflicted the burden on human health, and patients suffering from diabetic neuropathy, they are large in number. And of course, when it comes to molecular mechanisms of this particular complication, there are very few theories which gives us a clear idea about what is happening at molecular level when it comes to diabetic neuropathy. Now, there are many pathways which are activated during these disease conditions. So, pathology of this disease is linked with expression of many proteins inside the cell or even at the nuclear level. And there is a sufficient literature. The science has moved ahead to explore all these mechanisms in detail. And as far as this diabetic neuropathy is concerned, we focused on one pathway, which is the major molecular pathway taking place inside the neurons, that is WNT beta-catenin pathway. Now, this WNT beta-catenin pathway is expressed into the cells, leading to damage to the neurons. So we use this type 2 diabetes animal model, which consists of a low dose of streptozotocin, which is a toxic chemical for beta cells of pancreas, and high-fat diet. So high-fat diet along with streptozotocin, it causes type 2 diabetes, and if we will not give any type of treatment for the experimental animals, the animals will develop this type 2 diabetic neuropathy. So we gave treatment to all these animals for 16 weeks, and at the end of the experiment, we studied various biochemical parameters, then behavioral assessment, various inflammatory parameters, oxidative stress, histopathology, and we also studied expression of proteins at this neuron level, WNT and beta-catenin. Now, in each set of study, we took a standard drug. I told you that there is no specific drug for treatment of any complication, but the symptoms related with these diabetic complications, they are managed with the drugs which are already available in the market. So pregabalin, which is a known analgesic drug, we took for this particular study, and we used 2 doses of Vasanta Kusmakar Ras based on its clinical use. Now, coming to the results of this particular study, you can see that plasma glucose levels, they are decreased, and we compare them with diabetic control animals. Now, these diabetic control animals will not give any type of treatment. The same thing happened in case of glycated hemoglobin, which is one of the important marker in case of diabetes, there was a significant reduction in the glycated hemoglobin levels in animals treated with this VKR at 2 dose levels. Even insulin levels, then HOMA-IR, oral glucose tolerance test, all these parameters, they showed protective effect of this Vasanta Kusmakar Ras in diabetic animals. I mean, even in case of lipid parameters, we got significant results where there was a significant decrease in triglycerides, total cholesterol, LDL, and increase in HDL levels in experimental animals. Now, behavioral parameters, they are designed for this animal model, which are having similarity with that of humans, that a patient suffering from diabetic neuropathy will be having changes in the sensation. It may be change in the sensation related with heat or pressure, etc. So we tested the thermal nociceptive threshold levels by using hot plate. Now this is the first picture. You can see it's a hot plate where the plate is maintained at 55 degrees Celsius. So when animal is placed on the plate, it's a hot plate, you know, so the animal will jump or animal will try to withdraw the paw from that hot surface. Now this time is decreased in case of diabetic animals due to affecting the nerves which carries information to the brain related with change in the environment. In case of mechanical withdrawal threshold also, we could see that there was decrease in the response in case of diabetic animals, and we can see that there was significant increase in response time and the response pressure when the animals they were treated with Vasanta Kusmakar Ras. So this indicated that this is Vasanta Kusmakar Ras is having important function in protecting neuron, or it may be decreasing the progression of damage to the neuron in case of neuropathy. Now, we also tested nerve conduction velocity, which is also an important parameter when it comes to humans for diagnosing such type of condition. So motor nerve conduction velocity and somatic nerve conduction velocity, both these velocities were recorded with the help of a data acquisition system. And we can see that this VKR treatment, it showed significant increase in the MNCV and SNCV when we compared it with diabetic control animals. So as I mentioned, diabetic control animal, the motor nerve conduction velocity as well as somatosensory nerve conduction velocity will be decreased. All pathies, they are associated with inflammation. So all these inflammatory markers we could assess in the animals treated with Vasanta Kusmakar Ras or any standard drug, and we can see that there was significant decrease in majority of these inflammatory markers when they were treated with Vasanta Kusmakar Ras. We also assess this oxidative stress markers in neurons in this experiment. So we know that when there is inflammation, it is linked with oxidative stress. And when there is oxidative stress, again the organ will be damaged at higher level. So very important to manage this oxidative stress along with other mechanisms. And as the result, you can see that the oxidative stress markers like MDA, it was increased in case of all these treatment groups, including the standard group. GSS, SOD, and catalase, we can see that the increasing trend in all these important animals in the nervous tissue. Coming to histopathology, we could perform this histopathology of the sciatic nerve, which we isolated from all the experimental animals, and you can see that the damage to this sciatic nerve is less when the treatment group was examined for the histopathological changes. And at the end, as I mentioned, that WNT3 and beta-catenin pathway is the main molecular pathway in diabetic neuropathy, and we could assess the expression of WNT and beta-catenin in all these experimental animals. So it clearly says that WNT3 levels, they goes down after treatment with Vasanta Kusmakar Ras along with beta-catenin. So here we can say that this particular treatment, VKR treatment, improved this nerve conduction velocity and behavioral parameters, ameliorated the changes in plasma glucose level. It also improved the oxidative stress marker levels, and it prevented axonal damage, and it reduced the expression of WNT3 3A and beta catenin. So from all these results, we can say that, we can conclude that this formulation, Vasanta Kusmaka Ras, has significant effects in diabetic neuropathy via inhibition of WNT3 beta catenin pathway. Coming to the next complication, that is diabetic nephropathy. This is also one of the important complication of diabetes. Many a times it goes undiagnosed and the patient will suffer from chronic kidney disease or even end-stage kidney disease. So it's typically associated with arterial hypertension and increased cardiovascular mortality and morbidity. Now coming to this pathway, the molecular level pathway in diabetic nephropathy, TGF-beta, transforming growth factor beta 1 and SMAD, these two proteins, they are overexpressed in kidney in case of diabetic nephropathy. So we targeted this particular pathway and we propose that targeting this pathway, inhibiting this pathway will decrease the progression of diabetic nephropathy. The protocol was same. All the animals, they were administered with septozotocin and high-fat diet, and the treatment was given for 16 weeks. But the endpoint parameters were slightly different. We could able to study various urine parameters. Then different types of stainings were used to assess the changes in the histological structure of kidney, and inflammatory marker and oxidative stress also we could study. Coming to this kidney function test, as everyone knows that, plasma protein, urine protein. This ratio is very important. There was a decrease in the plasma protein levels of diabetic animals. And the levels were increased after treatment with Vasanta Kusmaka Ras. Albumin, which is one of the important protein, the same trend we could observe. And plasma creatinine and blood urea nitrogen levels were also decreased after treatment with Vasant Kusmaka Ras for 16 weeks. Now the important thing is that this is a type 2 diabetes study. So that's why the long-term treatment was selected. 16 weeks each animal was treated with a specific dose of VKR. And we know that it's not a chronic condition, it's not an acute condition, but it's a chronic condition which will require a long-term treatment. Now urine parameters also, we could get significant results like GFR, glomerular filtration rate, which is severely decreased in case of these animals suffering from diabetic nephropathy. And there was increase in glomerular filtration rate after treatment with VKR at 2 selected dose levels. Again, the inflammatory marker levels were also increased in all diabetic animals, and there was significant decrease in TNF-alpha, IL-6 and IL-1 beta, which are the specific markers in inflammation. There was a significant decrease. Antioxidant level enzymes, again same trend as that of neuropathy. We could see that all these antioxidant enzymes, they were increased in the specific tissue, that is kidney tissue. Now we studied structure of kidney at the cellular level by using different types of staining like HE staining, which is a regularly used staining in majority of histopathological studies. So you can see that there is increase in glomerular thickening when it comes to HE staining, and the glomerular thickening was decreased at the highest dose of VKR. So it says that the VKR is protecting all these tissues at specific level. Coming to this periodic acid staining, it is this is a special type of staining which is used for understanding the expression of glycoproteins. So abnormal protein, and due to abnormal protein synthesis, there is thickening of specific area like glomerular basement membrane thickening. It can be easily observed by using such type of staining. That is, we call it as PAS staining. And Masson's trichome staining, it is specific for fibrin, which is important protein in fibrosis. And as we can see here, the fibrosis, the blue color in the slide, it indicates the increase in fibrosis. But if you will see the picture of kidney VKR treatment at highest dose, 56 milligram per kg, you can see that very small amount of fibrin has been deposited at tissue level, which indicates that there is decrease in renal fibrosis. We also studied TGF-beta1 and SMAD in the kidney of all these experimental animals, and there was a significant decrease in this TGF-beta levels at both these doses of VKR, and even the SMAD 2/3 expression was decreased in all these tissues. So even you can just go through this diagram where, lisinopril was used as a standard drug in this study. Again, everyone knows that lisinopril is not an anti-diabetic drug, but it is an anti-hypertensive drug, but it is approved for its use in diabetic nephropathy. So we took this lisinopril as a standard drug, but as the results suggest that even the lisinopril is not significantly reduce this TGF-beta levels in the kidney. So at the end, we can say that VKR has significant effects in diabetic nephropathy by reducing expression of these two important proteins, TGF-beta1 and SMAD2. Coming to cardiomyopathy, it's a macrovascular complication of diabetes, and here we specifically took into consideration PI3K and AKT signaling pathway in cardiomyopathy. We also studied the role of GLUT4. GLUT4 is nothing but what? It's a channel protein which helps glucose entry into the cell. So obviously we'll be requiring more number of GLUT4 at cellular level to reduce the blood glucose level. And in diabetic patients or in diabetes, the GLUT4 levels, they goes down. The cell cannot synthesize GLUT4 at higher rate. Animal model was same, the treatment pattern was same, but there was slight differences in the parameters which we selected for this cardiomyopathy. Coming to this specific hemodynamic parameters, all heart-related parameters were studied by using data acquisition system. We could ligate this particular blood vessel so that we can record all these parameters by using data acquisition system. So heart rate, which was decreased in diabetic animals, it was significantly increased in the treatment animals at both the dose levels. Even PR interval, QRS interval, R wave amplitude, then systolic blood pressure, arterial blood pressure, all these parameters, they were improved after treatment with VKR for 16 weeks in diabetic animals. Now these are representative ECG graphs of experimental animals, that is Sprague-Dawley rats. Now coming to cardiac markers, CKMB and this troponin, these are two important markers which indicates damage to the heart or inflammation to the heart. And this CKMB and this troponin levels, they were increased in case of diabetic animals, the animals suffering from cardiomyopathy, and these levels were significantly decreased after treatment of this Vasantakusumakar rust to all these animals. Again, similar pattern we could observe for oxidative stress in heart. Even in histopathology, we could see that there is less damage at the tissue level in different types of staining. It may be HE staining, Masson trichome staining, even the fibrosis was decreased in diabetic animals treated with VKR. And in case of protein expression study, this PI3K, AKT, and GLUT4. You can see that all these important proteins, the levels, they go down in case of diabetic condition. And this VKR treatment, it showed significant improvement in the expression of these proteins which are important for the cell. So yes, this Vasantakusumakar rust showed significant effects in management of diabetic cardiomyopathy by activating these PI3K, AKT, GLUT4 pathway. Coming to this last microvascular complication, that is diabetic retinopathy. Again, there is no drug for management or treatment of diabetic retinopathy. The best way is to retard, to inhibit progression of all these complications by controlling different pathways. So here we took VEGF, vascular endothelial growth factor, and matrix metalloproteinase, that is VEGF MMP signaling pathway in retinopathy. And polyol pathway is also very important, which is related with this diabetic retinopathy, one of the important microvascular complication of diabetes. Now here the model is slightly different. As you can see, the rats, they were treated with streptozotocin, but 8 and 1, that is 9 weeks, they were kept as such. And after 9 weeks, these rats were treated with VKR and calcium dobicelate, which is a recently approved drug for management of diabetic retinopathy. It's not a specific drug, again, just a symptomatic treatment. 16 weeks treatment was given to each animals. So this was the, what you can say, longest animal model we used for studying this diabetic complication, that is diabetic retinopathy. And we could record electroretinograms of all these experimental animals. We did fundoscopy, biochemical parameters, histopathology, and studying the expression of VEGF and MMPs. Coming to this fundoscopy, as we can see that there is diabetic eye, it shows presence of newly developed blood vessels, angiogenesis. It's of course a marker in case of cancer also, but angiogenesis is typical in case of diabetic retinopathy also. So you can see the number of blood vessels, the increased number of blood vessels in the diabetic control animals. And the animals which are treated with calcium dobicelate and Vasant Kusmakar Ras at 2 dose levels, they show the less angiogenesis, which is very much required for controlling this type of complication. Then histopathology of retina, the hallmark of this condition is thickening of retinal tissue. And at the end, there is decreased in function of retina, which is very important for judging different types of colors through the visual pathway. So there was a significant decrease in the retinal thickness in all these treatment groups. Then we recorded electroretinogram of all these experimental animals. Now electroretinogram is nothing but what? Just like ECG, we study the pattern of stimulus and interpreting that particular pattern by using some software. So ERG study was carried out by using this data acquisition system. So you can see this setup of ERG study. The rat is in that chamber and the lights, they are switched on and switched off. So when the lights are switched on and switched off, the action potentially generated into these retinal cells and which is processed in the brain. So this particular processing of these actions is decreased in case of diabetic retinopathy. And we could record all these changes in eye at different light intensities. And all these recordings at different light intensities showed that there was a significant effect of this VKR when the animals, they were treated for a long time. So these are important parameters like A wave and B wave, and we could record ERG of each eye, that is left eye or right eye. So generally in case of animal experimentation, we keep one eye untreated and we just go for recording of all these ERGs in one eye. But we could able to record all these ERGs in both eyes in each animal. So this is the ERG pattern at 50 lux. So we can see that this A wave changes are prominent in case of diabetic animals, and the A wave pattern is increased in case of the treatment animals. So this is the data for 100 lux, this is data for 316 lux, and all these ERGs, they could clearly present the improvement in the structure of eye or decreasing the progression of damage to the eye. Then we studied these vascular endothelial growth factor and matrix metalloproteinase 2 and 9 in eye tissue, and you can easily make out that all these proteins, they are decreased in case of diabetic animals, and after treatment there was significant increase in these proteins which are there in eye of all these experimental animals. So from this study, we can conclude that this Vasant Kusmakar Ras has significant effects in diabetic retinopathy by suppressing this MMP2, 9, and VEGF proteins and improving the electrical conductivity of light into the eye. Now this work, we did this work to support this use of Vasanta Kusmakar Ras in preventing, or you can say prolonging, these diabetic conditions. So the present study work provides sufficient preclinical evidence that VKR treatment has holistic approach towards delaying the progression of diabetic complication, and the study provides a scientific support for framing a proof of concept of usefulness of this classical Ayurveda formulation in diabetes and associated complications. So this molecular level study will give scientific evidence for using Vasanta Kusmakar Ras for preventing diabetic complication in patients. Now this is my research team, all are my PhD students, and the majority of them, they have worked on various aspects in diabetic complications. Thank you everyone.
Thank you very much, sir. Dr. Yogesh A. Kulkarni, Associate Professor and Associate Dean at Shobha Ben Pratap Bhai Patel School of Pharmacy and Technology Management. Thank you very much, sir. And as a token of appreciation, I would like to invite Professor S.G. Ranasinghe, a well-respected and admired figure in the field of Ayurveda, to present the token of appreciation to Dr. Yogesh A. Kulkarni. Once again, a very big thank you for Dr. Yogesh A. Kulkarni for presenting the role of Rasashastra in diabetic complications and evidence-based revalidation. Rasashastra Sambhasha-6 edition. Ladies and gentlemen, thanks to Sri Dhootapapeshwar Limited, we Sri Lankans are privileged to have the benefit of Rasashastra nearly 15 years. Ladies and gentlemen, we, being Ayurveda physicians, we have to treat patients, not with a bunch of medication. Alpa matropayogitvat. Just simple tiny doses will do. So once again, thanks to Sri Dhootapapeshwar Limited, we do have the privilege of treating patients with safe, efficient, and even more with Rasashastra. So proceeding with the Rasashastra Sambhasha, the 6th edition, I would like to invite Professor Priyani Paranagama, the Chair of Chemistry at University of Kelaniya, Sri Lanka. Professor Paranagama is an eminent scientist with PhD in biochemistry from University of Glasgow. She is a member of the Executive Board in the Federation of Commonwealth Chemistry. Professor Paranagama has numerous research publications in indexed journals to her credit and also authored over 11 books. For her outstanding research contributions, she has been decorated by various awards, including the prestigious Dr. C.L. De Silva Gold Medal Award in 2015, Sosred Award from National Science Foundation 2017, Vice Chancellor Merit Award in 2021 and 2022, to name a very few. Professor Paranagama also held key positions such as the Director of Institute of Indigenous Medicine and now the Faculty of Indigenous Medicine, University of Colombo, the Dean, College of Chemical Sciences, Institute of Chemistry, and she was also the chairperson of the Formulatory Committee of Department of Ayurveda. Professor Paranagama would present you the unique insights of the standardization and characterization of Ayurveda Rasashastra. It's over to you, madam.
chairperson and the members of the head table, commissioner of the Ayurveda, Department of Ayurveda, and Mr. Ranjit Puranak, CEO of the Sri Dhootapapeshwar Limited, all the academic staff members, and doctors from Department of Ayurveda and also from other hospitals. The delegates from India and all the students from universities, indigenous universities in Sri Lanka and from India. Ladies and gentlemen, first of all, it's a pleasure and honor to deliver this speech standing before you on Rasashastra. I'm basically a chemist, and I have been working in the Ayurvedic doctors and also my fellow academic staff members in the Institute of Indigenous Medicine. So I was convinced and I made it to learn about the Rasashastra. And also I realized that there is a secret and also superior quality of Rasashastra that I need to work on it and convince myself. That is how I get into this Rasashastra. So with that, I'm today going to talk about standardization and characterization of Ayurveda Rasashastras. More than myself, you all know what are Rasashastras. They are made of herbal mineral or mercury or different mineral formulas. And this, if I name some of the metals that are present, maybe mercury, arsenic, tin, lead, so-called heavy metals. So chemists actually very scared of these metals. When it says heavy metals, they are so worried. I have experience in talking about arsenic. You all know that. So, but I thought that these medicine are given to patients, have been given to patients many years. We never died of one of these patients. So there is secret behind that. It is our duty to reveal that and expose to the general public and convince them. This is why I thought to talk about this Rasashastras. There are many important things. As you all know, mainly they are made with metal formulations, and also herbs also used to make them. But we know that this herbal material cannot be retained after preparation. And also these are given for chronic disorders. And what is important is method of preparation, which is utmost important. So there are many uses. Now the previous speaker revealed how these Rasashastras affect you on diabetes. Not only that, if you do a literature search, it says can be used for liver diseases, anemia, obesity, heart diseases, skin diseases, respiratory tract disorders, even cancer. And it is not ending from that, and also says the research have been carried out and prove that improve immune system, help to attain longevity, physical strength, mental competence, sharpness of sense organ. So work as rasayana therapy. So I gave attention to this chart. This made me, I was very excited to see this, the method of preparation. That tells us a lot of things. So starting from mineral, in Ayurveda language it says Shodhana, Bhavana, Marana, so the purification, and also you do wet grinding, heating, and also you prepare the bhashma. And when come to bhashma, that is the most important product which you have to prepare it carefully. We talked about nanoscience, but nanoscience is discovered long time ago. If you look at this, the bhashma, the particle size should be nanoparticles. Otherwise, I don't think you will be able to get the required quality. And also, these rasa aushadhis, if you look at, it is connected to this pharmaceutical, the bhavana is that Ayurvedic pharmaceutical method. It's connected to all these processes. And then, if you looked at the important factors that are related to rasa aushadhis administration, especially the dose. As it says, our computer says that the minute dose is used, the small amount that should be given, and also the duration, the time, and mode of drug administration, diet and routine management, shelf life. More importantly, testing and analysis before use the drug. That is very, very important, whether the drug is prepared in the way that to maintain the quality. So there are many advantages of these rasa aushadhis. As I said, use very small doses, and also easily palatable and fast acting. And we use incinerated minerals, possess excellent therapeutic values, and also they are very stable. Therefore, they have longer shelf life, and the preservation is easy, and the assimilation of rasa preparation is much more faster, because that preparation undergoes series of processes, and the efficacy of this rasa aushadhis was shown that increased with the time. That means you can keep it for a long time without expiring. However, there are problems. So all these rasa Aushadis, their purification method is well documented in this, the Ayurveda text, and also that purification procedure is given, how it should be used before administered into the human body. Unpurified or purely prepared rasa preparation can be highly toxic and also can kill a person. That could cause chronic toxicity too. Therefore, we know that the preparation procedure is not convenient, and also it is time consuming, and also we need to get a global recognition for this. Therefore, we need scientific evidence and also global availability, and need to report more and more scientific evidence in order to get the global acceptance. As you all know that there are 4 types of rasa aushadis. They are prepared in different methods. One is prepared by using mortar and pestle like Sitarama Vati. And also sometimes they are prepared as tin flakes. And also sometimes they are prepared inside a glass bottle, Rasa Sindura. And sometimes they use very special method. So that tells us where is the secret behind, and also where we should pay the attention when make these rasa aushadis. So I'm going to talk about little bit of knowledge I gain, and also my experience I'm going to share with you. The first thing, when you select the raw material, you need to be very careful, and you need to look at the physical characteristics such as color and shape, size, and its smell. So there are many things. All these are listed in the Ayurvedic text. And also, when come to preparation process, as you all know that the Shodhana is the most important, and also it's the first process in the preparation of rasa aushadis. That means the purification. It is very complex. So, as per the scientific method, if this process is done carefully, properly, so that help you to remove organic and inorganic impurities, and also help to reduce the particle size and material convert into herbomineral components, and that increase the brittleness. So therefore, that is one of the important steps in preparation of rasa aushadis. And secondly, you do this process called marana. So there you complete, what you do is complete incineration or calcification. So this, whatever the raw material that are taken is subjected to very high heat, and sometimes this range of heat that should be monitored. And due to this heat, whatever the material that are present, there are many changes take place. many chemical reaction take place inside. In other words, whatever you use metals, you convert them to their ionic form in the scientific way. So you prepare the bhasmas. So these bhasmas, the quality of the bhasma is the most important thing that you need to check. So therefore, characterization of these bhasmas is extremely important. These contain incinerated metals. And these are not new things, what I am going to talk about. All what I wanted is to connect this, the methodology that is stated in the Rasashastra text. We are now practicing. This is what I have realized. Using modern, sophisticated equipment. And prove that if we use the correct methodology to prepare the rasa aushadhis, you get the right product. Therefore, the physical characterization clearly laid out varna, the color. For an example, if you prepare the loha bhasma, it's like madang, or what do you call, I check the color to match it, it's like grape color. And also, if you prepare abra bhasma, then it comes like brick red color. So this is simple. You just check the color. But when it comes to qualitative test, even though these are qualitative, for me, I see a lot of science inside. So most of the tests, when I looked at it, I thought, so these are very super quality tests that have been given for this special medicine, which is a unique in Ayurveda. And if you do test it properly, then you get the right product. Let me start with Rekha Varnatva. So what you are asked to do is, take pinch of bhasma, you rub it using your thumb and the index finger, and then you see whether the particle embedded and whether they entered the finger furrows. What does it mean? If they can enter the finger furrows, if they are only nanoparticles. If not, you don't see them there, if the particle size are bigger than that. And also, the next one, you are asked to check the laksha, that is the shining. You take small amount of the, the bhasma product, and you see whether it is shining. If shines, then you don't accept it. That indicate the free metals are there. So these are very important quality tests that we have to go through. All these tests, when you look at slowly, repeatedly, you are asked to do it repeatedly, not just one. You start with one test, and also this is another test, and also this is, another qualitative test that called avami, that means this bhasma does not cause vomiting, no vomiting feeling. And also the next step with the flame, nirdumatta, that means this is very specific for arsenic containing bhasma, because if you prepared it properly and add into the fire, you don't see smoke. I just thought of my first year chemistry. We do flame test, you know. So where we take metals and mix with conc HCl and put to the flame. You don't see smooth because they converted to iron form. So here also, if metals, free metals are there, you see smoke. That means the quality is not good. We need to see the science behind that many years ago. And again, you are asked to do all these tests before prepare the drug. This test says, asking again, take small amount of the bhasma you prepared and put into water, like if it is floating, then only good. Again, you are asking to do and checked and confirm it's light. If the metal particle, they don't float, they just go down if they are free metal. If you see they are coming down, that means you have not prepared the drug properly. So, and also it says if you have properly incinerated and bhasma is prepared properly, it is tasteless. And more things given, they are chemical, under chemical characteristics. Asked to take some carbon containing material like ghee, jaggery, and equal amount you mix with bhasma, and you are asked to keep that in the crucible and subject to intense heat. Again, checking whether that's loosely bound, that is metal present or not. If loosely bondage of the bhasma can be checked by doing that. And also, what is this test with silver? You are asked to take a piece of silver and mix with bhasma, and then again to do the heat into intense heat. That again you are asked to check the bhasma is properly prepared. And if weight is not increased after adding silver, that indicate you have done or you prepared it properly. So that means the bhasma does not contain any free metals. So all what I wanted to, I know you all are expert in this field. I repeat, I thought like I'll tell you again to tell you again to convince you it is the methodology that we need to be really careful and do it properly. If you don't do it properly, then it can be really dangerous. It is time consuming, it's not easy, but it gives you a lot of good results if you properly prepared it, and also will be well accepted. At the same time, we all know that WHO guidelines for bhasma, it is given, and also when herbo-mineral formulations are prepared, or bhasma is prepared, all these tests should be done, and we check whether you pass, when the product is produced, whether all these tests you get good results or not. But even though the previous methods, what I said to you, we all convinced, I also looked into it, observed it, and carefully analyzed and convinced myself. But if you take the general public, it's difficult. They always looked at the new sophisticated methods and whether you use sophisticated equipment to prove this the product is in the acceptable form. So it is the new technology, and even among you also may be thinking, no, we have to do that also. Therefore, since these rasa aushadhis, they prove that their efficacy and also the effectivity, and also it is unique to the indigenous medicine. So the Ayurveda has a big, there's a big place for these rasa aushadhis. Therefore, we need to use new technology in order to prove the scientific community. If we prove scientific community, they will accept that. And we have to have this peer publications in peer-reviewed journals. So these are most of all this equipment I have listed can be used to prove whatever the formula. You know what formula of rasa aushadhi? Whenever that it is named, whether it is present or not. Let me take some of these equipment, and they are really important to prove the efficacy, prove the the structure. One is XRD. This is a modern sophisticated method that is used to analyze quality and quantity of the bhasma when you prepare rasa aushadhi. So this method is a rapid analytical technique. This helps to characterize the crystalline solid that you prepared, what you call bhasma, and also convince that it has the whatever that you have prepared. And next is the FTIR, Fourier Transformer Infrared equipment. This is also extremely important. This tells you what functional group present in your compound, and also then you know that the product you expected, when after processing, it is available. So therefore, now this FTIR is also used to prove the the product quality. And also SEM, scanning electron microscopy. This technique also important to look at the external morphology of the product you prepared. You prepare crystalline structures, and also to show that you have prepared nanoparticles. And also transmission electron microscope, TEM, that tells you the size of the particles and shape of the particles, and also, it is known that, say that mercuric sulfide is your bhasma. It should be present in your rasa aushadhis. So by doing this, you can confirm that it is there. And also ICP-MS. So usually the heavy metals content is quantified using atomic absorption spectrometer, but ICP-MS is available now that give the whole spectrum of metals available in your product. So therefore, by doing that, we can convince no impurities present other than the expected metals. Let me take one or two products and show you how it should be, share our experience, and when the rasa aushadhis are prepared, how you get the product and how you will see the spectrums and their profiles when you use this equipment. This is You all know that Rasasindur, which is red sulfide mercury. So it also prepared using mercury, sulfur, and also with herbal product. And then you make the kajali and you get the, the final product, which contain the, thermal treatment of black sulfide of mercury, because you introduce different temperatures, final product is obtained. And then after getting the final product, you can see the organoleptic properties, that is, the like touch, that the Ayurvedic test very clearly given to you. This is what you have to do to see whether, nanoparticles are formed or not. Similarly, we can do the test I mentioned using this equipment. Let me show you. This Rasasindur powder was used and get the XRD. This is how it looks like. And what you do, so it contains this mercuric sulfide, it is known, and this profile is made and available, so that can be kept as the standard, and whenever you prepare it, you can do XRD and compare with this one, whether it's properly prepared or not. Whether if additional peaks present, what is meant by that? That means impurities are present. So therefore, this is the mercuric sulfide nanocrystals. So, and then the TEM. So this the first diagram that shows the low magnification image, that shows the presence of nanoparticle. And when the B picture, it is high resolution TEM that shows the, the detailed diagram of these crystals. And C, that is diffraction pattern. These are given in nanoparticles. And also, you can look at the particle size distribution graph that can be prepared and that can be kept as the standard for this special medicine. And EDX shows what metals, what elements present in your rasa aushadhis. It clearly indicates mercury, sulfur present. And also you can use X-ray photoelectronic spectra, again that indicate presence of mercury and sulfur. And also Raman scattering spectrum, very clear spectrum can be obtained for mercuric sulfide. And also absorption pattern can be monitored using optical properties. And this can be used as a standard, and whenever produced, we can see that whether its pattern is available or not. Then if so, then it is reproducible. Otherwise, it won't be in the same quality. This is another example, that is preparation of anilari rasa, which is a herbo-mineral compound. These are the ingredients that were used to prepare this, and then prepared the bhashma, and also prepared this classical Ayurvedic rasa aushadhis test to see the quality, whether the nanoparticles are present. In this, there was an additional test was done, that is Dadi Pariksha, because copper is used in this one to prepare this rasa aushadhi. This is very specific test for copper, when copper is used. So this bhashma is sprinkled and see whether you get the blue color. If blue color present, that means you fail, right? If no blue color remain, that means you have prepared the Bhashma properly. Similarly, SCM was done, and detailed diagram is given here. And also XRD is done. Here it contains copper sulfide, mercuric sulfide, and both are present. And also EDS shows very clearly only mercury, copper, sulfur present as the elements. And also the last experience I am going to share with the research that is conducted by Professor Harapaddeniya, that is with Lokanatha Rasa. So she's an expert in the Rasa Aushadhis area, and her project is to look at the hepatoprotective activity of Lokanatha Rasa with special reference to hepatocellular carcinoma. So the drug was prepared, and these are the ingredients that were taken. And also the prepared using the prescribed method in Rasa Aushadhis text, and then the test using this local test to read this text and see whether it has been done properly. And also, it contained different Bhashma was prepared, and then prepared the kajali, and also then mixed with the beetle juice and heated to very high temperature, and this is the product that was taken. The final preparation, ash color, smooth powder, and it was checked very well and see whether the product is formed. But we did not stop from that. We again did the characterization using our new modern equipment, like we did the particle size analysis, and also we use ICP-MS analysis to see whether mercury, calcium, those are the two, and also the sulfur availability. And also we looked at the FTIR analysis, and we wanted to do HPLC analysis to see whether any organic compound retain or not, and SCM analysis, XRD analysis. This is XRD clearly says that calcium carbonate is present, and FTIR, so that prove that carbonate groups are present. The other side, all the peaks are unique to carbonate bonding, carbon-oxygen bonding, and the HPLC says that no peaks, that indicate no organic compound, all gone. And also, after doing that, she will be presenting, you will see her work, but we did some biochemical work as well. We wanted to look at the antioxidant assay. We use antioxidant activity using DPPH assay, and also we test ABTS antioxidant further using ABTS assay. And also we have done the ORAC assay to check the antioxidant, and we found the IC50 value, that is the inhibition 50% inhibition concentration is very, using a small amount that can be, the IC50 values are comparatively small. And also further anti-inflammatory assays and antibacterial assays. Those things were also done. So therefore, these Rasa Aushadhis, there are many work we have to do, and also we need clinical trials and prove that their efficacy, and also we are hoping to do metabolomic studies. That means after giving these drugs, administration of these drugs, what changes that you see in the blood, that can be done. and also whether some of the, the compounds that appear in the blood can be controlled, and those are like the future research that we wanted to do, we are planning to do. So, and also the toxicity studies that you will hear from Professor Herapaddeniya. So with that, I conclude my speech. And first and foremost, I thank the organizing committee for giving me this opportunity to present this talk on Rasa Aushadhi, standardization, characterization of Rasa Aushadhis. And also I thank the Sri Dhootapapeshwar Limited for organizing this conference and inviting us, and also Department of Ayurveda, especially the Commissioner and the Department of Ayurveda for collaborating with them, and my colleague, Professor Herapaddeniya, joining with me and doing this research. I really appreciate and thanking her. And also, because this is the way to go to prove the efficacy of that. And also my own university, thanking them, having them the facilities and also the research lab to conduct this research in the university. Thank you very much for your kind attention.
We invite Professor Paranagama to remain on stage, and thanking you, Madam, for being with us and delivering this wonderful speech. And I invite our chair of the forum, Professor S.G. Ranasinghe, to present this more memento and the token of appreciation to Professor Priyani Paranagama on behalf of Sri Dhootapapeshwar Rasashastra Sambhasha. Thank you so much. And the next session is going to be by Professor S.K. M.K. Herapaddeniya, Head Department of Ayurveda Pharmacology, Pharmaceutics and Community Medicine, Faculty of Indigenous Medicine, University of Colombo. And she has done MD in Rasashastra from Banaras Hindu University, Varanasi, India, and currently pursuing her PhD from the University of Kelaniya, Sri Lanka. With enormous experience in the field of academics, research in Rasashastra, she would be presenting the topic of safety and efficacy of Ayurveda Rasa Aushadhi. Over to you, ma'am.
Good afternoon, respected chairperson, co-chairpersons, respected professors, delegates from India and Sri Lanka, Department of Ayurveda, officers from Department of Ayurveda, all respected invitees. It's gone. Okay. No, first. Okay. my topic is safety and efficacy of Ayurveda preparation. I think in Professor Parnagama Madam's presentation, we all get some idea how to prepare a quality rasa preparation. But after preparation of this rasa aushadhi, we have to know how we can safely introduce this preparation to the human. Rasashastra is the main branch of Ayurveda pharmaceutics. We know we use herbal, metal and minerals for our drug preparations. But herbal preparation always we discuss because most of our preparations prepared by using herbal. But we have to consider especially about how we use these metals and minerals for our diseases. For different preparations, how we efficaciously and safely use for different conditions. That in Rasashastra is the branch which explain all these metals and minerals how safely used for different disorders. Actually, in our previous presentation, Priyani Madam correctly told these metals, minerals and these herbo-mineral, all these preparation considered as rasa aushadhis. Why? We all several times discussed why all these metals and minerals preparations are superior, because we use in this preparation in very tiny dose, and these preparations not giving any unpleasant taste. That means these preparations are very palatability, and its shelf life also having long shelf life, and efficacy, it's having good efficacy. So that's why we considered these aushadhis are more superior preparation than herbal preparations. But we have to know all these metals and minerals are heavily toxic. So how we can convert these toxic materials into non-toxic pharmaceutical condition? Yes. Now globally there are very serious questions arise for the rasa aushadhi, because these rasa aushadhis contain severe toxic materials like mercury. That's why always they raise questions, how you give this preparation to the human being. We face many problems, especially indicate this rasa preparation for different disorders. But we know all these preparation used from thousands of years from our Ayurveda physicians. But what is the secret between this preparation mean? In Rasashastra followed different processing techniques to prepare these materials into non-toxic form. Always, you know, Ayurveda, we not consider about the curing of the disease. Always we have to preserve, or we have to prevent the disease. It is the most valuable thing. Prevention is, I think, in Swasthasya Rakshana is more valuable than the curation of the diseases. So that's why this rasa preparation we can use for preventive and curative and also for the rejuvenative actions. Always, when after preparation of this rasa aushadhi, we have to ensure the safe use of this product, and we always try to avoid the occurrence of unwanted effects to the patients. It provides information on possible health hazards likely to be avoided from repeated drugs over a limited period of time. And also, we have to ensure this safety of this study, always we use toxicity study. For this toxicity study, initially we use different animals. we can use rats, rabbits, and for monkeys. For in this studies, very simply we can use rodents because then after we can progress these toxicity studies to a clinical trial after when we complete the toxicity part. In this study, especially I'll give you attention regarding the toxicity of Lokanath Rasa because this study we not completed yet. This is also ongoing research. Already we completed the standardization part of this preparation and also some antioxidant effect and anti-inflammatory studies already we completed and toxicity study also completed. We have to do a clinical trial for this preparation. This Lokanath Rasa also a mercurial preparation. It is categorized under a Potali Kalpana. Paranga Madam told there are 4 types of preparation mentioned in Rasashastra. Among these 4 preparation, this Lokanath Rasa comes under Potali preparation. It's a special preparation mentioned in Rasashastra. These are the ingredients of Lokanath Rasa. It contains Shuddha Parada, Shuddha Gandhaka, and equal amount of Shuddha Parada and Kandhaka convert into a Kajjali, and 2 parts of Abraka Bhashma, 2 parts of Lauha Bhashma, and 2 parts of Tamra Bhashma, and 6 parts of Varatika Bhashma include in this preparation. After preparation of Kajjali, all these Bhashmas mixed together with Kajjali, and then this mixture Bhavana with 7 days Bhavana with beetle juice, and then subject it to Sarava Samputa, and finally give to Gajaputa, and we can obtain Lokanath Rasa. It's of ash color, very fine powder. The recommended dose for this preparation is 2 to 3 Rattika, and it is, we prepared this Lokanath Rasa according to Rasendra Sara Sangraha. If you go through Rasashastra text, there are many Lokanath Rasa preparation mentioned in Rasashastra text, but we selected this Rasendra Sara Sangraha Priharoga Adhikara reference. And this preparation mentioned in Ayurveda Pharmacopoeia also. Then we selected the topic hepatoprotective activity of Lokanath Rasa with special reference on hepatocellular carcinoma. This project funded by the United Nations Industrial Development, UNIDO Geneva, government of Switzerland, through Ministry of Environment, we got the grant. And this is the study design for this preparation, because I'll come to the safety study. Before come to the safety study, I'll briefly mention the study design. First, the preparation and analyze the physicochemical parameters of this preparation, and then safety evaluation, acute and chronic toxicity studies, then evaluation of in vitro antioxidant and anti-inflammatory activity, 4th, evaluation of the anti-carcinogenic activity of Lokanath Rasa by using hepatocellular carcinoma cell line. Finally, clinical assess to assess the effectiveness of Lokanath Rasa for non-alcoholic fatty liver disease. Here, now I'll describe how we conduct the toxicity study of this preparation. Even before start the animal studies, we have to get the ethical clearance. We got the ethical clearance from University of Peradeniya Postgraduate Institute of Science. Here I selected male and female viscerats. Their weight between 150 to 200 grams. 6 weeks old rats I selected. We purchased this rat from MRI, and the oral toxicity study was performed accordance with the Organization for Economic Cooperation and Development. Here, young, healthy, non-pregnant male and female rats we divided, we randomly selected, and 6 animals in the one group, 5 groups we used for this study. Then each group of rats are separately housed in a polypropylene cage in ventilated room. Ambient temperature 22 centigrade, relative humidity 40 to 65%, and light 12 hours dark, 12 hours light photo period, and they were provided with rodent food and purified water. All the animals acclimatized to the laboratory condition at least for 7 days prior for the experimentation. I conducted this animal toxicity study in Faculty of Medicine, University of Ruhuna. Here study group, in one group there are 6 animals. The dose for the acute toxicity I gave 200 milligram per kg body weight by using oral gavage, and animals were closely observed initially every 4 hours within 24 hours for acute toxicity. But no observation of treatment related death or toxic signs, lacrimation, lethargy or abnormal respiratory pattern, salivation or chronic or tonic movements are not observed within these 24 hours. Then the repeated dose or the chronic oral toxicity study I conducted because always we prescribed these rasa preparation in a very short period, but I done this chronic toxicity study for 90 days. Continuously I induce this drug for 90 days. Here study group 30 rats, and these rats also different doses I introduced to this rat 90 days continuously. I gave 5 milligram kg per kg body weight, then 15 milligram, that means 3 time dose, and the 25 milligram 5 time dose, 10 time dose, 50 milligram per kg body weight. Likewise, different concentration of drug I introduced to different group rats. After 90 days, the animals were sacrificed, then for their hematological, biochemical and histopathological studies conducted. In this study, I used specially liver, kidney, lungs, spleen and intestines were extracted, weighed and stored at 40 centigrade in 10% formalin. Slides were prepared for histopathological studies. These are the histopathological studies. These histopathological slides I took from the high doses treated group. That means 10 times treated group histopathological studies. Now you can see here male kidney slides and the lungs and the small intestine, spleen, heart and liver. When we compare this histopathological slides with control group, no any significant deformities noticed, because these slides read by an eminent pathologist in University of Colombo, Faculty of Medicine, and the pathologist in University of Peradeniya, Faculty of Medicine. Both of them see these histopathological slides and know any abnormalities observed in these histopathological slides, even in kidney, lungs, small intestine, spleen, heart and liver. I observed their food intake. Once a week I recorded their food intake. We didn't observe any abnormal things because they taken their food in usual manner. Water intake also, weekly I observed, and there is no any abnormalities in their water intake. If you can go through these values, you will not see any abnormalities in their water intake. If they are in a ill condition or some unwanted things happens, there this food intake, water intake will can some different values, can get some different values. You see their body weights. Body weight is normally they gain their weight. I think after completion of the 90 days, they are in a very healthy condition, no any weakness we observed. Here we express our data likewise, and we not see any abnormal things in their body weight, water consumption, and also for their food taken. Then after sacrifice, I weighted their organs. We not observed any abnormal weight gain in their organs, especially if we notice some abnormalities. The abnormalities mean if any toxic effect happened to the organs should be in edematous condition. The organs weight should be increased. If you go through the weight of these organs, we not observe any abnormal weight in their organs. This is the hematological analysis. We collected blood in heparinized tubes, examined using an automated hematology system at a commercial diagnostic laboratory, and these are the results in their full blood count. Total white cell count, neutrophil, lymphocyte, monocyte, eosinophils, basophils, hemoglobin, RBC, this hematocrit, and MCHC. We need not show any abnormal values in their hematological parameters also. That means oral consumption of Lokanath Rasa had no undesirable consequence on the circulating blood cell as well as on their productions. This is their serum biochemical analysis. I collected their blood in a sterile tubes without any anticoagulant coating and allowed to stand for 30 minutes. And then samples were centrifuged at 1500 gram for 10 minutes at 4 centigrade. Still further processing. Then we done the biochemical testing. These are the biochemical assays: blood urea, serum creatinine, SGOT, SGPT, alkaline phosphatase, and blood sugar. No undesirable consequences on biochemical parameters. After seeing these toxicity study values, my recommendation is when we prepare the mercurial preparations in standard way, we can safely introduce this drug for our human body for different disorders. For further study, actually we done this study for anti-carcinogenic activity on Lokanath Rasa on hepatocellular carcinoma. I think hepatocellular carcinoma is one of the most dangerous and the most commonest carcinoma, especially in Asian countries. Nowadays, we facing many problems because non-alcoholic fatty liver very soon, without any alarming, can convert into a hepatocellular carcinoma. That's why we decide to evaluate the anti-carcinogenic activity of Lokanath Rasa by introducing different doses to the liver cancer cell lines. Here I selected for this study HepG2 cell lines. We purchased it from United Kingdom Cancer Cell Lines Institute, and then we evaluate the effectiveness of Lokanath Rasa. These are the sample was prepared as 20 milligram per milliliter. Cisplatin we use as a standard drug because cisplatin is a very common standard drug for cancers, especially for liver cancers. That's why I used cisplatin 0.1 milligram per milliliter were used as the positive control of the study. These are the results. Then 4 milligram concentration, we notice it's having anti-carcinogenic activity, but we know we have to when we conduct anti-carcinogenic activity by using cell lines, after we determine if having the anti-carcinogenic activity, we have to use this drug on a carcinoma stem line, because the drug should affect on the stem cells. That means it should be a effective drug for cancers. Some drugs can destroy the cancer cells, but if this drug is not potent to destroy the stem cells, the drug will not work. So we have to see how this drug acts on cancer stem cells. That's why now very recently this supervisor of this project is Madam Priyani Parnagama. So Madam advised me to purchase a stem cell line and the liver stem cell line, cancer cell line, to further do the studies to the stem cell line study. So I think for our next research forum, we can see how our drugs act on cancer stem cell line, especially liver cancer cells. And also, the further studies, we will do the genotoxicity study. By using this cell line, we can do, because there are many arguments related with mercury can cause several genes damage. I think it's a very common argument in society. That's why we want to see the genotoxicity of this product, especially the mercurial product, how acts on genes. For this study, I'll do a COMET assay and also for micronucleus assay. By using the COMET assay, we will decide how it acts on our genes, and by using micronucleus assay, we can see how this drug acts and how it will do the DNA breakdown or not. I think it may be a valuable values or the valuable study for not only limited for the Lokanatha rasa, it's only for the whole mercurial preparation. When we consider the safety of our rasa preparation, always we have to practice GMP. Good manufacturing practices are essential to produce quality, genuine medicine. And in Rasashastra, especially toxicity and other unwanted things are the concern, always concern. Therefore, this toxicity can happen when we use the improper usage of raw materials. That's why Parnagama Madam emphasized how we select drug by considering the acceptable features. Then, therefore, methods of raw material authentication and standard operating procedures like Shodhana, Jarana, Marana, Amrutikarana are recommended and we have to follow according to our Rasashastra text. And furthermore, quality control parameters of finished products such as Bhashma Pariksha also we have to follow, and then it's also hold in an important place. Finally, I tell also this formulation have specific guidelines for usage such as Patyapatya, dose, Anupana and Kala. So always we have to consider these things, and then we can use a very safe and efficient preparation for our patients without any doubts. Thank you very much for your kind consideration.
Thank you, Professor Harapadenia, for this informational session on Rasaushadhi, the safety and efficacy of Ayurveda Rasaushadhi. And we invite you to remain on stage as we have a small token of appreciation, a memento presented by co-chair of today's forum, Dr. S.L. Vebalvala. Doctor, we invite you to present this memento to Professor S.K. M.K. Harapadenia. Thank you, Madam. And we also invite you to note down your questions that you have in order to get the answers, get everything clarified from our honorable speakers, our lecturers, and our speakers who are delivering their presentations, the sessions, the scientific forum at today's Rasashastra Sambhasha, the 6th edition of it. ඔබ වෙනුවෙන් ඔබේ ප්‍රශ්න සඳහා පිළිතුරු දෙන්නට අපේ දේශකයන් සූදානම්. මේ කතිකාවසානයේදී ඔබට අවස්ථාව ලැබෙනවා ඔබට පැන නගින ගැටලු ඔවුන්ට ඉදිරිපත් කරන්නට ඒවට පිළිතුරු ලබා ගැනීම සඳහා එනිසා අපි ආරාධනය කරනවා ඔබ පොතක් පෑනක් අරගෙන ඒ ප්‍රශ්න ටික ලෑස්ති කරගෙන තියාගන්න අවසානයේදී එම පිළිතුරු දැන ගැනීම සඳහා. And our next session would be by Dr. Mangalacharya, who is an alumnus of SDM College of Ayurveda, Hassan, India. She is a clinician with a successful track record of over 20 years. Patients from around 15 countries, including Japan, Colombia, Romania, USA, have been benefited from her Ayurveda treatment expertise. She has in various forms across the globe and created awareness about facts and about Ayurveda through her articles in various magazines. Dr. Acharya specialized in Garbha Sanskar from Garbhopanishad, Jamnagar, and would share her significant experience on integration of Rasaushadhi with pregnancy. Over to you, ma'am.
त्यांचा नाही त्यांचा काही संबंध नाही तिकडनं येत नाही एक्सप्लेन प्रेझेंटेशन होत नाही आहे. हे अपडेट आलं ना त्याच्यानं जोर झाला. दे चालू कर चालू कर चालू कर.
get cured fast. So they have no time to wait. And second condition is ama. You know, ama is, as you all know, I mean, from Ayurvedic community, it's a very common condition because of our mithya ahara, mithya vihara. So we have no time to give them ampachana and different methods to start treating. So easy way is rasaushadhi, but I don't give rasaushadhis in all conditions. In emergency conditions, we have to go for it. And one more thing is we have competition now. Before we were not recognized. After COVID, during COVID, people started recognizing us. Okay, they came to know that, okay, something is there. There is one more thing, we can go for it. So we have to be ready to give them. So when they are coming, when the people are ready to come to us, we have to have all the weapons or whatever we have to be with us so that we can treat them, we can cure their, heal their problem. Yeah, especially during and before pregnancy. So I have taken only before conception and during 9 months of pregnancy, my experience in these days. So first of all, this why I have chosen this means. So most of the time before pregnancy, they have taken lot of medications and mostly these women will be taken will have taken this pills for a postponement or the contraceptive pills. All these are we can consider as Garavisha. So Garavisha is very difficult to treat. They have to undergo Panchakarma, Shodhana, all these treatments. And also as all our previous speakers mentioned, it is easy and palatable. They have no time now, as most of them, I come from Silicon City of India. So both the husband and wife are working in IT field. Some are in day work, daytime, some at night. So it's very difficult for them to meet. So they have no time to take the treatment also. So these are all the Rasaushadhis are the gems for us when we treat them. But still I follow Shastra. I tell them to undergo Shodhana. Then only if we start the medicine, it is more effective with the clean system. So that's the reason. And also the chances of getting during pregnancy, if I give kashaya or different churnas, and it's very difficult for them to swallow. First of all, they find it difficult to eat normal diet, their food. And on top of that, if I give them bitter medicine, they can't swallow. So this is one of the reason. And most of the time, I don't give Rasaushadhi during pregnancy. Only in emergency conditions. Some of the examples I'm going to show in my future slides. Most of the time, I try to give them with the Kastaushadhi or with the Ahara, I try to treat the problem. With the A2 ghee, A2, you know, the indigenous cow's ghee and indigenous cow's milk, that is one of the best medicine we can give during pregnancy. In ancient times, when the lady gets pregnant, they get a new cow to the house because most of the households had cows. And it was like, if you are rich, that means you have more cows. So that I'm talking about indigenous cows, not the present cow, which is not very, the milk or ghee of this cow is not very healthy. So those cows, and now still we have, but very few cows we are left with. So we have to improve the our farming of the indigenous cows. It is Bos indicus, I think so. Yeah, Bos indicus, which otherwise the new the thing is called. It is Taurus. So that is not good. So with those things, I try to treat the patients. But in different situations, which is emergency, we have to go for Rasaushadhis, and that too good company we know, so I'm confident and give these medicines. So, and we know why Rasaushadhi, so till now all the speakers were mentioning what are rasa, we have Uparasa, Maharasa, the forms, and they are very quick acting, and the side effects also we have to think about it, and we consider patients as but most of the time they act very good, but behind our back they continue taking medicines for longer duration, and they try to give it to others in their family. This is Ayurvedic medicines, you can take, it has no side effect, and they start, they become their own, become Vaidya and start prescribing to others. So we have to be very careful while we give Rasaushadhis. It is not that it gives some side effect, but as Acharya Charaka says, Purusham Purusham Viksha is a very important part in our science, which we are not, most of the time we don't follow. So anyone we give this medicine, so which is not good. So before giving any Rasaushadhis, we have to not how mercury side effect, all these things we cannot explain to them. At least we should tell them this medicine, Rasashastra, you have to take for this duration only. We have to make sure, otherwise once they take the medicine, they take it home and they buy it from some shops and they go on taking it. That we have to be careful. So little bit of, at least we have to give them the knowledge of the, if they continue, what are the things which may happen to them. Nowadays they're all Google gurus. They go search in the Google and they find out more than us. They start asking questions. So this is also one thing we have to be very careful, and we have to be patient to explain to them how to not to believe these things and trust the Vaidyas. So before that, Vaidyas should have more trust on the science. And I feel if we have all our Brahatrais, Lagustrais, if we follow it properly, we get very good trust. And on top of that, we have our scientific community who are doing a lot of research. With them, we can definitely proceed and continue with our Rasashastra. About the usage of Loha, I was searching for Rasashastra in Brahatrais. I couldn't find any of the Rasashastra in the Brahatrais. I thought I could find two opinions in that. First one is maybe they are very healthy, because when we go for this Itihasa, we can see Atreya when they went to Ira for the good health. That means they were all healthy. They wanted good health to do more tapasya and these things. Very, I mean, like Rasayana they were expecting, and later Jwara came, all different diseases. But maybe they were very healthy, so they were not, they didn't want to go for Rasashastra. Especially during pregnancy, Rasashastra were not mentioned in our Brahatrais. Only we can find is in Pumsavana Vidhi of Charaka Samhita, because I follow Pumsavana Vidhi not for as a male child or something, to have a Shuddha, Atma Shuddhi of the fetus before 10 to 12 weeks of pregnancy. Tomorrow is a Pushya Nakshatra, Sunday and Monday this week. So I have to be there for some procedures I follow during Pushya Nakshatra. We know Pushya Nakshatra the day where all the drugs or all the medicine values will increase, potency of the drugs will increase. So that time if we administer, we get the good results. So that time he has mentioned Tatha, here he says Kanakamayan, Rajatamayan, also Ayasascha, Loha and Kanaka and Rajata, gold and silver. Purusha Agnivarna Purusha, and when the statue should be prepared, a small idol should be prepared in these metals like Kanaka, Rajata and all. And it should be kept in milk, ghee, dadi, it should be, and then that water, that liquid should be taken, prakshipya, pibe, pibe danasheta, Pushya, on the day of Pushya Nakshatra, the lady should soak these, only in those situations, metals are mentioned for pregnant women. And also in Ashtanga Sangraha Sharirasthana, first few weeks of pregnancy, when she gets the Garbhagrihita Lakshana, she has to take care of Garbha, because chances of abortion is more in the initial period. So at that time, they have given these metals as rasayana, that too metal, liquid or milk or ghee should be kept in the metals like Sarpi, Shaliparani, Palashabhyam, Shrutam, Kanaka, Rajakata, Kwathitam. So they should be kept in the Rajata or Kanaka, and Shailodaka Anupana Pivet. So that should be Shitodaka, that Shita Udaka should be kept in these things, Kanaka, Rajata, and kept, and they should drink it. Not, they have not mentioned any preparations. Only in Yogaratnakara we can find Rasanjana and Gairika Prayoga, that too during the, if it is like a threatened abortion, Garbhasrava we can find. Otherwise, no mention of these herbs and these Rasashastra. And one more conclusion I feel, that that time herbs had very good quality than present day herbs. So later on, these acharyas, like later came this Nagarjuna and all, they have mentioned more about Rasashastra during pregnancy and even other conditions. Initial textbooks, we can't find much of the Rasashastra. Then common Rasashastra I use before conception. So before conception, I don't care. You know, people, they don't, I mean, it's not contraindicated or anything. So most of the time people come with, as I mentioned earlier, they don't follow proper lifestyle. Ahara, vihara, and everything is mithya, and they're not ready to accept also. So I have to, from the day, like what time you are getting up. So I can't tell them to get up in Brahmi Muhurta. At least make some changes and going to toilet. They sit there for longer duration. That we have to correct. That will also help in, like, it is like apana and mala. It's too much of without their knowledge they'll be straining and that toilet. So that is also not good. From there I have to correct. Again Abhyanga, at least once a week we advise them Abhyanga. Those things should be corrected, then only we can treat them. I tell them initially, so Ayurveda you have to follow, 50% is in your hand, 50% is in my hand. I can't do a magic and treat you. So most of the time they come to us after undergoing all the treatments, so they'll frustrated and they'll listen to us. So some changes I make initially, and when I see the condition, it's like PCOD and most of, and their, some of them are having fibroids, so they have to undergo panchakarma, especially kapha prakriti, I do vamana and virechana, otherwise just virechana. And sometimes if they have no time, just a sadhyo vamana and virechana. After that I start these rasaushadis along with the other kastaushadis also. So 2 months, 3 months, we can see lot of difference in their conditions. Even AMH level changes, and this hirsutism and all these things are very common nowadays because of, mostly I see, because of their lifestyle. So I always tell, I make fun of them. You dress up like a woman, you act like a woman, then you can have the estrogen hormone. Otherwise you will not have, you will have testosterone. I tell, because many women don't like me because I talk like this, because I can see the difference. Here in our place, we do, once the girl gets her menarche, we do a lot of functions. I am from southern part of India, where because they dress up like a, make her like a Devi Ma, like a Durga, and Lakshmi. That goes into there, it is like a muscle memory. Then she thinks she is a woman. She dresses up like a woman. Otherwise, she won't dress up like a woman. The way her action, everything is very important. From there, we have to correct their lifestyle. Mostly, many people, they say they just don't like me because of the way I speak. So that is where we have to correct. Then the bodily changes happen. Even mind, body, everything has to change. And they should be calm. They have to become calm before taking any medication, because pregnancy is not easy. Nowadays, we are finding so much of mental and physical problems in the children. So I tell them, you have to sacrifice some things in your life before getting pregnant. And till 30, 34, 35, they're busy planning other things. And if later even then they don't decide whether to have children or not. It is too late. How many eggs were remaining? Because of the wrong lifestyle, most of the eggs would have gone. So those things we have to educate them. So for that, Rasayana Chikitsa is very important, and Shodhana is important. So Hemagarbha Pottali is one of the best, it's my favorite thing. That too, they have this Vaidya Seva. From there I order and I use, and especially during COVID, it saved many people. 60, 70 oxygen level, we rubbed it in we have that stone with the potali, so made people to have, lick it, or some of the doctors, even allopathic doctors, they've made them through the tube, they passed it, and they could bring them back from 60 to 90 oxygen level. And many, it's a miracle drug, I can say, and I give it in many cases. Even in small children, I give Hemagarbha Pottali, so as Rasayana, and with the even previous abortions. Some people, they abort the child or fetus, or some will undergo a natural abortion or some other health problem. So definitely that Visha will be there in the pelvic and that Apana Marga. So we have to cleanse them. Then the Rasayana Chikitsa, we give them this. And even in some cases where they go for IVF and fundus thickness is not attained, then they come to us. We do first one virechana, after virechana, 1 month or 2 months, this Hemagarbha Pottali or some other, along with that like Falasarpi or some Grita, that will improve their uterus size and nourishes the uterus. So then they go back and they have conceived. And usually there's a previous history of abortions. Before conception, 3 months, my Garbhasamskar procedure, 3 months before conception, we give brahmacharya for both. We tell them to follow brahmacharya. Both mind and body, brahmacharya is very important. We tell them to have all sattvic food and sattvic lifestyle. We can convince them, but we should take some time and explain to them how our ancestors, we have to give them some examples, and also we should tell them how bad the situation is going now. So during that time, if they have any history of previous abortion or family history of even autism and different, like the first baby is having some problem, for second baby if they are coming, after Shodhana, I advise Hemagarbha Pottali for at least 1 or 2 months. So that will help in, I had just two cases I had. Previous child, one had some ADHD. It's a very hyperactive child. The second child, now she's 2, she's fine, all her milestones are very good. So after giving Hemagarbha Pottali, this helped me a lot. And this Rajatagarbha Pottali also, the Rajatagarbha Pottali acts as Garbhashaya Shodhaka. It helps in fibroids or this PCODs and endometriosis. All these things I start after Shodhana. I don't know if it might be helping if without Shodhana, but mostly I give this with after Shodhana only. At least because some of my Western patients where I cannot give them the proper Shodhana, Sadhyo Virechana at least I tell them to have like 2 days some Amapachana, where whatever is available in their place. Then I tell them to do one castrol cleansing. I just name it as castrol cleansing. Early morning take some 20 to 3 m according to their Agni castrol, and till afternoon they'll clear the system. Then after that after 2 days I tell them to start. Depending on their condition, I advise these pottalis or other medicines. And Pushpadhanvarasa in PCOD and endometriosis, and that's what Bija Shuddhi. If it is unknown cause is also we know unknown cause is the main cause for not conceiving. Both the couple, Pushpadhanma is also very good. And Stri Vyadhiharasa is Anubhuti Yoga from Dhootapapeshwar Limited. I am writing this regularly. For young girls also, PCOS is very common for 16, 17. Now we can see this condition. So for even irregular menstruation, mostly nowadays the pelvic area is very weak for a woman. That is the main part which is weak, and I tell them to do exercise because in just 20, 30, 40 years ago, they used to carry water in India. They used to carry water from the well. They used to sit on the ground and cook. Nowadays, nobody sits on the ground. That squatting position itself is gone. They can't even bend their legs. So there is no circulation to the pelvic area. I advise them to do this sit-ups. I advise them to sit on the ground and at least while studying or doing something, at least half an hour per day, sit on the squatting position, and application of oil on their pelvic areas, lower abdomen, back, every day, at least twice or thrice a week, and having this ginjali or tila kashaya, lashuna, tila regularly in their diet or extra. So that will improve their apana and as well as their pelvic. That area has to be improved. Because of that, nowadays we have PCOS very common in young children. So from there we have to correct their lifestyle. And Abrakabasma it's also as a rasayana we give, and people with always like frequently their HB is less. 10, 9, so whatever we do, it will not come up. So that time with the proper Agni Deepana, with the lifestyle like Abhyanga, I advise Abrakabasma along with the Sitopaladi, some combination of Churna, every day I advise. And Suvanamalati is I always tell Vasantamalati is my favorite in most of the condition. Whenever I get confused, Shotha, any kind of Shotha in the body, it acts as a miracle drug. Just today also I got a call saying it's fine now, because in Bangalore at present so many dengue cases and it's high temperature, 104, 105, and people, they are so used to Ayurveda now. Whoever comes to me, they never, whatever is the emergency condition, even last week 1-year-old baby, fine. No?
Right, right. It's same the slide?
Same.
Slide, it's
Yeah. So what is happening, I don't know. Maybe I touched something.
this one?
Yeah,
Next,
yeah.
Next slide.
Yeah.
Right?
Yeah, fine. Sorry. So 1.5-year-old baby, 104 temperature, they are not ready to go to allopathy. So started with Mahalakshmi Vilasaras initially, and temperature came down, but still phlegm, and the baby is having problem. So went for this Swarnamalati Vasanta. So whenever I get confused, and less dose, not that I am giving in a full dose, so that will help like anything. Even for after Jwara, they feel very tired, they have to go to work, and no time to recover, like long-term recovery. So Vasanta Malati, you take for 10 days with some diet, and I make sure that they should stop the medicines. If they continue, I don't think anything will happen, but still I won't allow them to continue. So these things, in emergency, it will help. So it is actually out of topic, still I remembered, so I told. So Punarnava Mandura. So Punarnava Mandura is my favorite drug for pregnant women. Usually, if everything is normal, I would not suggest any medicines throughout 9 months. With the diet and with we have all, like each month we tell them to have different like first 3 months, we don't worry much with the Grita and Kshira only, because most of the time they have this nausea and digestion is not very good. So with Madhifala Rasayana, and we only concentrate more on Agni. Preservation of Agni is very important for first 3 months. So with these, if they don't take any other supplements during first 3 months, they will not have nausea. Mostly I have seen some of the, even they have some family stress and other things they have. Otherwise, chances of getting nausea is less if they are not taking any supplements. I would not suggest any supplements to them. And with the ahara only, they should improve their health. After 3rd, 4th, 5th, I start with some basic aushadis, like if at all they need, Punarnava Mandura is the best one. Along with that, I tell them to have this raisins, and in diet also certain changes. So and it improves. One of my patient had 8, her HB was 8, and her gynecologist suggested for, because many of my patients, they take all the supplements or medicines, everything from me. Just for checkup, they go to gynecologist, because she's the one who conducts delivery. I'm not supposed to do. So all other things, they come to me. Only for delivery, they go there. So 8 was her HP level, and she came to me, running to me, and Punarnava Mandura twice. First few 10 days, I have given thrice a day. Then I reduced to twice, and within a month, it was 9.5 or 10. Then she told them, yeah, I have taken all the injection and everything. It improves like anything. Even 10, one of them had 11 also. So it increases very fast. And even for edema, and chances of getting after 6th and 7th month, this pedal edema and chances of getting PIH is also can be controlled, because Punarnava is also Mandura, it also Shotahara, and Pravala Pisti and Kamaduga, Mukta Pisti, all these are very commonly I use for Chardi, as well as it acts as because some of them have this tendency, they want some iron calcium. Instead of allopathy, they want to come to me. So I tell them it is not needed, but still to keep them happy, and also it is not bad to give these things. So Pravala Pisti, or according to their condition, or this Mukta Pisti I give. And we have a folklore system in my place in Karnataka. We give this coconut flour 3rd, 5th, 7th month of pregnancy. This is the folklore system. What I do, I add some of the Ayurvedic herbs, Pravala Pisti and Mukta Pisti I add according to their this thing. 3rd month I add Arjuna for that, as the Hridaya development takes place. 5th month Panchame, this Chetana, so I add some Brahmi. And 7th month overall development, so Shatavari, and like just I add along with whatever the coconut flour and this kesari, so I add this. So this is the folklore system, and with the Ayurveda I've added. And Garbhapala Rasa for only if they have any spotting or anything, I suggest Garbhapala Rasa. And also even previous history of abortion, Phalasarpi or Garbhapala Rasa, I want them to continue for at least 3 months. And one lady, she came to me and at her 37. So she had 3 abortions before that. So for her, after Shodhana, 1 month of Hemagarbha Puttali, and during pregnancy, first 2 months, Phalasarpi, and 1 day Phalasarpi, because she didn't want to take Sarpi, so 1 day this Garbhapala Rasa, and now she has a second child is 8 months old now. And Manasamitra in case of sleeplessness and stress, because they'll be working along with when they're pregnant, they won't take leave and they have to manage at home. Because of their sensitivity during the pregnancy, they get irritated, they be fighting and shouting. So Manasamitra, some cases I add. Manasamitra Vatakam and Swarnavasanta Malati. I can show an example of Swarnavasanta Malati in one of the case. So any Shotha and Chandraprabhavati, most of the time people, women are getting UTI during pregnancy. So in the milder cases, I go with Chandraprabhavati. Even also it is in PIH, so pregnancy-induced hypertension, and some people just they have hot flushes. Pitta Vriddhi Lakshana in pregnancy, Chandraprabhavati helps a lot. And also it is like Shotha, any like pedal edema and conditions. Even Punarnavadi Kashaya helps a lot for them. And that's what Rasatantra Sara and this Siddha Prayoga Sangraha is the book which very like they have given all the most of the Rasaushadis during pregnancy. So that book is a very good one to refer for this type of situations. But still I tell not to give just without any reason. I would not suggest. Only they have any problem or history, anything, let's go for Rasaushadi. Otherwise we have good caste Aushadhi. Gruta is one of the best thing we can give during the pregnancy. Different Grutas at different months also we can go for it. And this is the case study I wanted to share. She is the 42-year married for 2 years. I had a session last June. I had a session from Sitaram Ayurveda. It's like international thing. Many came from outside. She listened to me. She's a MD Ayurveda doctor. She wanted me to mention about this. And so that's what I'm telling. So she was married 39 or something. And after 1 year, she didn't conceive, so she went to a gynecologist, and she is a Kapha Prakriti person. Naturally, she is at the heavier side. So when she went to the doctor, as usual, it's a common protocol, so weight loss. She had to undergo weight loss. So before that, she herself took one Virechana for herself. After that, she went to gynecologist and started with the raw vegetable diet and intermittent fasting and this millets. So all Vata Vardhaka, Vata Pitta Vriddhi, Ahara Vihara, and vigorous exercise, everything she started. And within 3-4 months of treatment, she didn't get her this periods, and she went for the checkup. So then she came to know all her values that I am going to show were high. And along with that, she had hirsutism, she had dysmenorrhea, whatever the symptoms. Vardhakya, she was I told her, it was her Vardhakya. When she came to me, she was at that stage. So this was the treatment I decided. Initially, I had to explain to her to follow Vagbhata, Ashtanga Hridaya, first 4 chapter. This is like, we have to make sure before advising anyone, we should follow at least some part of our Samhitas. Ashtanga Hridaya, beautifully he mentions. We ourselves tell our patients not to take rice, and this is like, it is not our science. The procedure of preparation of rice is different. Rice is not bad. So we have to study these texts first and follow, then we can advise to the patients. So this is the plan. I started because of the Rukshata, whole system got Rukshata, and Vata Pitta Vriddhi, she attained her menopause. So start with the change of plan. Diet is rice she has to have. She is from Andhra Pradesh, where that's the highest they grow rice, and that is their diet. And preparation is they have to remove the starch from the rice when she eats, and ghee in the rice, and exercise is must, abhyanga is must, and matra bhasdi, because the rukshata of the body. First I started with Sudshekha Rasa as Ampachana, because definitely she needs some Ampachana. Then matra bhasti and Phalasarpi matra bhasti. After a week, Hemagarbha Potali I started, and Palasarpi every day, along with Sudhshekara, because we should not get Ama, first of all, Kapha Prakriti person. So then she continued it 2 months. This was her value of AMH was like 0.01, and her FSH is 30 something. 33. So it was in June when she came to me. So this is August. So because when they called me, I had cases ready. So she had small follicles. She got her period. She called. Before that, she called me saying that she's feeling light. She has put on some 2-3 kgs. I told her it is her normal weight. No, not to worry. And she got her periods. Dyspareunia was lost. She was enjoying her sexual life. Then she got her small multiple. This follicles could. This just before last week only. I went for this her test, and this is her latest report. I know this month she will not have good follicles, so next cycle definitely she will improve. So this is and she told me to mention about her profession also, because Ayurveda, being Ayurveda Vaidyas, it's our duty to follow at least 50% of trust our textbook, then definitely we can succeed. And this is the Rasashastra during pregnancy. This is also last month only I got it. So she came, it was like emergency condition. Her husband was literally carrying her. She had all the symptoms of dengue. She couldn't walk at all. All her limbs were swollen, and she was just came to know she was pregnant, and the doctor told her, and her in the lab report, typhoid was positive, but she had symptoms of all her dengue symptoms were there. And she was literally crying. She has a baby of 1.5-year-old child, and she is already 6 weeks pregnant. So we started with Shadangapaniya Muhur dose. Usually with this type of emergency condition, I tell them to have Shadangapaniya Muhur dose, and then with that I started with the Suvarna Vasanta Malati for 1 week. So this is the pregnancy test report. So this is after the 1 week of test, the Kashaya, they went for pregnancy, again reconfirmed, because they got scared. Something, after a Saushadi, something bad would have happened. So this is her test positive. This C-reactive is 18.5, both the typhoid tests are positive in this. So after, in August, you can see this. So just both are negative. So that too in 9th of August, on 9th of August, this last month. So within this time period, all the values, before the values, all her symptoms were, she was symptom free, and she was very happy, because she had to take care of the first child, and she has to take care of herself. So even last 2-3 days ago, she went for anomaly scan. So all her these things are fine. So this is the third case. So last year, I even now I haven't seen this lady. I got a call from a person. He's working in a DRDO scientist, and he's from Bihar. Bihar is very far from my Bangalore. So they called because when her 11 weeks anomaly scan showed no nasal bone. So they told them to abort the baby, this fetus. So he was searching, and he could see Garbhopanishad Foundation, Jamnagar. He called them. They said, as you are in Bangalore, contact. So they gave my contact number. So he was in tension. I told them, wait for 4 to 5 weeks, then go for the abortion. He said, my wife is then she'll get attachment with the child, and it is very difficult to. I said, give me 4 to 5 weeks. At least I can help you. So I didn't see her. I sent the medicine with her husband. So this was so we can see no nasal bone, and the marker, so 2.18. So that was the lab report. I got it. Then after 8 weeks, they had a scan. We can see no structural congenital malformation detected. So now it's a baby, 1.5-year-old baby is there. So just this Balamula Kashaya, and that she was not taking it because she was feeling nauseating, because Kashaya during pregnancy is difficult. Only with Hemagarbha Pottali, for one whole Pottali she consumed for 6 to 7 weeks, and then they went for the scan. These are the 3 main cases, because recent cases I could see. And also I have forgot to mention this fetal arrhythmiasis also we can, with the Kshirapaka also, Yashtik Kshirapaka it will help. Also Hemagarbha Potali helps because I have treated many of these cases. Some people don't turn up. Once they are fine, they just call me. I tell them to send the report. They don't bother. So I don't have many these things before and after. So, but we can treat many such conditions as it is fast acting during pregnancy. It acts fast, and even for these fetal anomalies, Rasaushadis can help us. And I have many other examples also in pediatric cases and other things. But this is the main part where we can give very healthy mind and body, very healthy children to the society from our Ayurvedic side. So thank you, Dhootapapeshwar, for calling me, and thank you, Sri Lanka.
Thank you very much, Madam. Dr. Subha Bangalacharya, who is an alumnus of SDM College of Ayurveda, Hassan, India. And Madam, as a tribute, we have a token of appreciation from Sri Dhootapapeshwar Limited. So I would like to respectfully invite co-chair of today's event, Dr. K.P.K.R. Karunakoda, Senior Lecturer and Head of Department of Shalya Shalakya Prasuti Tantra Kaumara Brutya, Faculty of Indigenous Medicine, University of Colombo. It was an insightful presentation which we significant experience on integration of Rasa Aushadhi with pregnancy, as we all are afraid to treat Rasa Aushadhi with such conditions. So thank you very much, Madam, once again. She deserves a big round of applause from the audience. So we learned more practical insights from you, Madam. So a gentle reminder for the audience once again, keep some notes. So we have an open session, open for you all, so you can pose questions. සියලු දෙනාටම ඉතාමත් කාරුණිකව නැවතත් නිවේදනය කරනවා පොතක් පැන්සලක් ඇති. ප්‍රශ්න තිබෙනවා නම් කෙටි සටහනක් තබා ගන්න. විවෘත සැසිවාරයක් තිබෙනවා. එහිදී ඔබ සියලු දෙනාටම ඔබ සියලු දෙනාගේම ගැටලු निराකරණය කර ගැනීමට අපි අවකාශ සලසා දී තිබෙනවා. ශ්‍රී දූතපාපේශ්වර ලිමිටඩ් විසින් සංවිධානය කරන 6 වෙනි වෙළුම රසශාස්ත්‍ර සම්භාෂා. So ladies and gentlemen, moving ahead with the proceedings of today's function, 6th edition of Rasashastra Sambhasha, I would like to invite Dr. Priyadarshini Gunaratna. She is an Ayurveda consultant in surgery, currently working for Gampaha Vikramarajchi Ayurveda Teaching Hospital, Yakkala, Sri Lanka. She has done her Masters in Shalyatantra MS from SDM College Ayurveda, Karnataka, India. And Dr. Gunaratna would present her rich clinical experience in successful management of critical wounds with Rasa Aushadhi and other Ayurveda medicines. Over to you, Madam.
From here I have to go. Ayubowan and very good evening to all the dignitaries on the dais and off the dais, and all distinguished guests in the audience and all the participants who are presenting here. I welcome you all for my presentation. So I think you all have experience with wound healing. No one can tell we are not having any experience of wound healing, because while we are birthing itself from the mother, while we are detaching, we are getting a wound. So it is very important to know what is the wound and how it's healing, and especially in
It's your presentation.
yeah, but it's previous one.
another experience in, especially in India and Sri Lanka, that all girls in Sri Lanka and India, and in India in boys also, they are having Karna Vedana Samskara. Mostly we are thinking that is only for that beauty purpose. No, that is not beauty purpose. It is actually for, we are introducing Agantuja Vrana for the body, and we are giving chance to resolve the wound by itself, because we are normally giving that golden jewelry to the ear or the nose. So because of that, it is very important to talk about wounds. So today I am going to deliver successful management of critical wounds with Rasa Aushadhi and other Ayurveda medicines. So this is the topic summary, that introduction of wounds, wounds management, common use in medicines, successful stories, summary and conclusion. Then while we are talking introduction of wound, especially in Susuta Samhita, they are given that it's very painful condition of the life, and most of the wounds appears on the skin. The same time, that skin is the largest organ on the body, and wound healing is the major problem for especially surgeons as well as other doctors also. And that especially 3 words they have mentioned in Susuta. That is Roga called Vrana, that who is having Vrana, they call Rogi, that is Vranita, and where the patient is staying when they are getting wound, that called Vranita Gara. Then stages of wounds. Especially while we are treating for the wound, we have to consider about the stages of wound. That is mostly Drishta Vrana, that means that is not clear wound, and there is no any progress of wound, mostly it has stopped itself and no any granulation. Then Shuddha Vrana. Then Shuddha Vrana means they are not having slough or pus or any pain or any other symptoms, and they will start to grow the wound. Then it's turned to Vruhyamana Vrana, that is growing wound, and next one is Ruda Vrana, that means almost healed wound. Then what are the basic principles to manage wound? As a surgeon, it is very important to plan the wound healing first. Then we have to do the Vedana Shamana, Shotahara, Rakta Stambhana, Ropana Karma. Then we have to consider about Patyapatya, and mostly we have to do that prevent recurrence, because lot of patients, they are getting the same wound at the same site in various time. Because of that, we have to stop the doing recurrence on the body. Then here I have going to mention, this is Shashtivida Upakrama mentioned by Charakacharya, and Susuta Acharya, and the Charakacharya has mentioned 36 Upakramas. Here specially I have mentioned because they are having more Upakramas, but I am not going to detail one by one, but we can do a categorization of these Upakramas, what they are going to do with this. Then here that management of Vrana Shofa, they have given some ideas. Then incision and drainage, that means the Shashtivida Upakrama, how we can use while we are doing that Vrana Chikitsa. Then that cleansing and promoting granulation. For that also they have given some karma, and that other one is set right to wound healing and that Vaikrutapaham. For that 5 categories, normally we can contribute all these 60 kramas. Then while we are treating the wound also, we have to follow the normal protocol of Ayurveda treatments. For that, we have to go for Langana Karma, then Agni Deepana, Amapachana, Shodhana, Shamana, Tarpana, and Rasayana Karma. Especially while we are using that Rasayana Karma, we can prevent recurrence of wounds. Then what are the common medicines used in Vrana Chikitsa? Then here again I am telling we are not always using all rasa drugs, but we are using that other herbal drugs also. But specially I am going to mention what are the special rasa drugs we can use here. Then Agni Deepana and Pachana, we can use Abraka Bhashma, Kapardika Bhashma, Yasada Bhashma, Agni Kumara Rasa, Agni Tundi Rasa. In here we are using some Sri Lankan drugs also, mostly that Ramabana Rasa, then apart from that Navaratna Kalka, etc. Then Krimihara, especially that most of the patients having some macrocrimi or microcrimi. At that time we have to use Krimihara drugs, especially Trivanga Bhashma and Krimikutara Rasa also we have to use. Then Virechana, especially while we are treating the patients, we have to do the blood purification, if not the Rakta Shodhaka. For that purpose, we have to go for Virechana karma. So, most of the patients, while they are having wounds, they are having associated complaints, mostly they are having chronic constipation or malabadha. So, because of that, we have to use that Virechana drugs, mostly we can use Icha Bedi Rasa, then Agni Tundi Rasa, Arogya Vardhani Rasa, etc. Then because of the wounds, they may be having pain, then may be having Shota, then may be having Kandu, etc. According to that also, we have to give some medications. If the patient is having Daha, then we can use the Chandrakala Rasa, then Punarnava Gugulu, etc. Then Shotahara, especially that lot of patients, if they are having wound, they may be getting Shota. Then at that time we can use Amuttadi Gugulu, Singhada Gugulu, Panchamruta Loha, Gandhaka Rasayana, etc. Then especially that, that for Shulahara, because lot of patients having severe pain of their wounds, because of that we have to use that Shulahara drugs. The same time we have to do for that Shotahara treatments also. At that time we can use Trivanga Bhashma, Krimikutara Rasa, Punarnavadi Gugulu, Chandraprabha Vadi, etc. Then Jarahara, lot of patients they are having inflammations because of their wounds. So at that time we have to treat for that also. Then Abraka Bhashma, Punarnavadi Gugulu, Gandhaka Rasayana. In here also we are using some kind of Sri Lankan traditional medicines also, some kind of Ayurveda medicines also, apart from these. Then Pandu, most of time the patients may be having anemic conditions associated with the wounds. If the patient is not having good hemoglobin count, then the wound will take lot of time or more time to get healed. Because of that, we have to prevent the Pandutta stage of their wound. Then for that we can use Yasada Bhashma, Arogya Vardhani Vati, Punarnava Mandura, etc. Then most of the patients, they are having wounds because of their Madhumeha condition. If not, they are because of the diabetic conditions, especially diabetic foods and etc. For that also, we have to treat the same time. Then we have to give that Trivanga Bhashma, Triphala Gugulu or any other diabetic controlling drugs. That's what I want to mention. Not only the rasa drugs, that other medications what we are using here, either traditional or Ayurveda medicines, we have to give and we have to cure the diabetic as well with the wound healing. Then Rakta Shodhana, I told earlier also, if that blood is very good in condition, the wound heal very fast. If not, we have to do the Rakta Shodhana karma also. For that, we have to give Rakta Shodhaka drugs. And other thing is Rakta Stambhana. Some patients, they are coming, especially that cancer wounds, they are coming with lot of bleeding. And some patients, while they are having their Bruhyamana Vrana, they are having bleeding full of the wound. At that time, they will somewhat afraid. So we have to stop the bleeding. And mostly post-op, that means after the surgeries, they may be getting severe bleeding. At that time also, we have to use that Rakta Stambhana drugs, especially we can use Bola Bhadrasa, then Trivanga Bhashma, then Yashada Bhashma. Likewise, we can give lot of medications. Then Shofahara, that means inflamed wounds. For that also we can give that Amurtadi Gugulu, Kanchanara Gugulu, then Chandraprabha, then Gandhaka Rasayana, Panchatitta Grata Gugulu, etc. Then Kanduhara, especially that earlier also I told, lot of patients having Kandu while the wound is healing. At that time we have to give some Kanduhara medicines like Amurta Guggulu, Punannavadi Guggulu, Gandhaka Rasayana, etc. Then we are just go and have a look what kind of ingredients we have that we are commonly use, that some kind of rasa drugs. Here I am not going to mention one by one, but you can see in one drug they have lot of ingredients. Because of that lot of ingredients, they may be getting lot of actions. So as previous madams and all said, then from one medicine we can treat for lot of problems in a one time. That means mostly they will time saving also, they will prevent from recurrence also, they will prevent from suffering also. So here also you can see from one drug they are having lot of ingredients as well as lot of actions, and we can use from many conditions. In that Kapardaka Bhashma, especially I want to mention here, apart from these all, with my experience, lot of that Shravi wounds, that means discharging wounds, we can use that Kapardaka Bhashma. Lot of time, because of the Shrava in the wound will take more time to heal the wound. So we have to stop that Shrava, including Rakta Shrava and other all Shrava, that means maybe pus or serous or whatever the Shrava. So at that time we can use Kapardaka Bhashma for the wound also, that means locally, and the internally also we can use Kapardaka Bhashma. Then Trivanga Bhashma. Here also what I have to mention here, that they also having lot of actions. According to the patient's situation and the associated other complaints, we can use all these medications. Then Yashada Bhashma, especially where we can use Yashada Bhashma locally, while the wound had started their granulations. We can stimulate the granulations by putting that Yashada Bhashma on the wound, that means local application. So it is very beneficial treatment for the wound healing. Then Shanka Bhashma, the same as Kapardaka Bhashma previous I mentioned, that Shanka Bhashma also we can use for that Shravi wounds for the local. Apart from these, actually they have given that internal actions, but I am specially mentioned what we can use for that as local medications. Then Kukkutanda Twak Bhashma. This also we are using because some patients having fractures, because of that only they are getting some wounds. If not, maybe they are coming with osteosarcoma, some patients I got, some having osteomyelitis patients. At that time they are having very we need to do that bone density and all increase. So at that time we can give that Kukkutanda Bhashma specially. Then Mukta Bhashma also we can use for make that bone density correct. That Vanga Bhashma also likewise we can give. Then Agni Kumara Rasa. That previous also we mentioned, most of the patients while they are coming for us, they may be having armor condition in their body. If we are not doing that Nirama Chikitsa, that medicines will not work on their body or in their body on locally. So we have to convert that armor patient to the Nirama condition. At that time we can use Agni Kumara Rasa specially. In that Bolabadda Rasa means some patients I told earlier also may be having severe bleeding conditions because of post-op conditions or because of some other reasons. At that time we have to stop the bleeding as soon as possible. Because of that we can use that Bolabadda Rasa, and it has already Vrana Ropaka and it can enhance the immunity also. Then Agni Tundi Rasa also having Deepana Pachana actions. Then Arogya Vardhani Vati also we can use in many aspects. Then Icha Bedi Rasa, that also we are using for that Virechana purpose, because some patients having very chronic constipation, sometimes they may be having motions for once in a week or once in a month even. At that time we have to use that Icha Bedi Rasa and it will work very nicely. Then Krimikutara Rasa, that also either internal krimi or external krimi or either macro or micro krimi, we can use that Krimikutara Rasa also. Then Gandhaka Rasayana, that also very wonderful medicine, because most of the time if the patient having any inflammation condition, especially cellulitis and other reddish with fever and all other conditions, at that time we can use Gandhaka Rasayana, especially after the surgeries. While the surgeon has made any wound, then our responsibility to cure that wound as soon as possible without recurrence and without suffering or without complications. At that time, we can use the same day also that Gandhaka Rasayana, combination of other medications. It will give very good benefits to the patients. Then Amurta Adi Gugulu, that also very good for actually in inflammation conditions, especially associated with fever and redness and likewise and severe pain conditions. Then Kanchanara Gugulu, that also giving very wonderful results, because while we are using Kanchanara, that wound will getting smaller very fastly and it will prevent some bleeding and all other conditions. So we have to use that Kanchanara Gugulu also, but always I am telling, it is not only we are giving alone that medicine, it is a group of medicine, but it will have very good role for wound healing. Then Lakshadi Gugulu, that also, because lot of patients I am having because of fractures, they are having non-healing ulcers for long time. At that time we have to treat the fractures also. At that time especially we have to use Lakshadi Gugulu also. Then Punarnavadi Gugulu, that also giving very wonderful results because of that chauffeur, that means inflammation. If not that shota, that means they are having swelling. Sometimes they are having fever, sometimes they are having hypertension or other conditions. For most of the conditions we can use that Punarnavadi Gugulu very nicely, and we are getting very good result while treating. Then Singa Gugulu also having lot of, I think you can see lot of actions. According to the situation, we have to select the group of medications, then you can treat the patient nicely. And that, see the Chandra Prabha, I think I couldn't get able to finish all the ingredients also from this slide. That much ingredients they have, and they have that much lot of indications. So we can use in lot of places. Likewise this Chandra Prabha, not only wounds, even in a lot of places we can use very nicely and very successfully. Then Pachanamuta Gugulu, here that also mostly we are using as just Shamana and Tarpana Avastha. That means in the beginning, it means while we are doing that Shodhana Karma, we can't use that Pachanamruta Loha Gugulu, but while we are doing Shamana and Tarpana time, we can use this for especially blood purification and enhance the blood quality. In Panchatitta Grita Gugulu also, we can use the later part of the wound healing, that means in Ruhyamana Vrana. Then Trayodeshanka Gugulu, that also you can see lot of these medicines, they are having anti-inflammatory actions and analgesic actions, and I think you have seen some that lot of medicines, they have that combination of actions. Because of that, we can use that all medicines in rational way as a combination, then they will give very good result. Then Tripala Gugulu, that also very popular medicine while practicing surgery, because most of the post of that means after the surgery we can do that, we can give that Tripala Gugulu to past wound healing, and the same time we can use at mild laxative also, antimicrobial also. Likewise, such a good actions they are having, and we can use in correct places. Then Chavana Prashavaleha, why we are giving this? Because the patient must be in good Tarpana condition. For that we have to give that Chavana mostly in the Tarpana stage. Then these are the success stories I have treated. That's what I wanted to discuss with you. This is the wound. Actually, it came, I think, before 2 years. That was the pricking wound. After that, that other hospitals, they have done instrumental debridements, and they have tried 2 months, and the wound was worsening day by day. Because of that, they have given date for the amputation. But after coming to me, you can see that 6 months what I have done, according to the good combination of good medicines in a good rational way, so that patient save his leg. This is mycetoma, that means that common name is Madura foot. I think you must not have heard this much. This is actually the indication of amputation. Most the other medical systems, they are not having any medication for this. Only the final decision for this is amputation. For that also, I have started actually with Uddartana, then followed by Achapana, then Virechana Karma, then I started Shamana Karma, and the same way I have local treatments also done, especially what I have early mentioned, that Kapardaka Bhashma, likewise I have used. The meantime, the later parts I have used Rogani Kash, and later I use that Yashada Bhashma, etc. Anyhow, within 2 months he cured, and I think it was before 4 years. Still the patient is coming to me for the follow-up. No any recurrence up to now. This is necrotizing fasciitis. This also very severe case, because this also for indication of amputation. This patient also approached, I think, after 4 or 5 weeks later that started. Because of that, our Ayurveda wonders, and especially Ayurveda medicines, I was able to treat, and he was cured that much from 4 months treatments. And this is sphenoidal sinus. This was a very wonderful case, because she has undergone 6 times surgery, but the 7th time she approached to me, that means for Ayurveda treatments. At that time, she got cured within 3 weeks of that wound, but she told that last 6 times she suffered more than 3-4 months for wound heal. But while we were doing with Ayurveda treatments, she got healed that much within 3 weeks. But daily I have done dressing, including Matra Basti and local dressing, as well as internal medications. Then cellulitis. Most of the time that cellulitis, that other medical systems, they are very afraid to treat, and for this patient also they have given the date for the amputation for this leg. But that patient approached to me, but within 3 weeks he got very nice result after Ayurveda treatments. Then venous gangrene. Actually this patient was approached to me for his fistula case. I don't know whether you can see or not. In this side, he having one open. While we were treating for 1 week, at that time he developed that venous gangrene condition, but within 3 months, all Shastra Karma and all other karmas I have followed and got all the good result. And one thing I want to mention, I have never used any allopathic medicine, antibiotic, any painkillers. All the cases I have treated only with Ayurveda medicine, with Ayurveda wonders. And this is a diabetic foot also. This also given the date for amputation before 3 years. That also, while she was coming, it was full of stuff and pus, and just only cavities on her right foot. But after 9 months, she got very nice result, and up to now she is very well, and she is coming for the follow-up. And for her, that diabetic condition also, I gave medicine, and it was very nicely managed. Then osteomyelitis case. This patient also approached to me around February this year. At that time also they have given the date for the amputation for this patient. For this patient also having full pus and slough. At that time I started Kapardiga Bhashma internally, and around the wound I put that Dashanga Lepa with tamarind leaf juice likewise, and internally I have given a lot of medicines in rational way, but while 3 months he got that much relief. And the summary, what I want to mention, most drugs having large number of ingredients, especially I mentioned here that rasa drugs, then each drugs having lot of actions. So some single drugs can use for many conditions. Then the minimum number of drugs gives various effects. Then correct combination of drugs gives more benefits with minimum time duration. With same drug can manage complications also. Cost effective treatment can implement, and very less side effects and harmful drugs interactions. Then conclusion, a wound can be managed successfully with 60 upakrama if chatuspada of therapeutics are ideal. Then wound will heal within short time when chikitsa methods used in proper time, in proper order with proper medicines, can prevent complications, amputation and recurrence with using of Ayurveda wound healing methods mentioned in Susuta Samhita, especially I mentioned as a surgeon. Thank you very much for the chance I have to, you given me to present this presentation. Thank you and have a nice day.
Thank you very much, Madam. Dr. Priyadarshan, Consultant Ayurveda Surgeon of Gampaha Vikramarachi Ayurveda Teaching Hospital, Yakala, Sri Lanka. So, Madam, for your valuable insights presented here at the 6th Rasashastra Sambhasha, we would like to felicitate you, and I would like to cordially invite co-chair of today's forum, Dr. S.L. Vevalvala, the Senior Lecturer and Head of Department of Ayurveda Basic Principles at the Faculty of Indigenous Medicine, Gampaha Vikramarachi, University of Indigenous Medicine. Thank you very much, Madam. And while listening to this tremendous lecture, I found the memory of one important sloka. Nigudanukta Leshokta Sandigdhartha Pradipika Sunischitarth Vibudahai Paribhasha Nigatyate. Thanks to Dhootapapeshwar Limited, we got the opportunity to share this immense knowledge that would be beneficial for us to proceed forward as successful physician. So we saw insights that allopathic medicine, which we majority believe cannot solve, but Ayurveda, once done progressively, the allopathic treatments cannot be managed. Yes, we were able to manage. We have a tremendous medical system. We can believe, as one keynote speaker mentioned, yes, being doctors, being Ayurveda physicians, at least we need to have 50% of belief on our own medical system. So, ladies and gentlemen, it's the 6th Rasashastra Sambhasha. Gentle reminder once again, we will be having an open forum. Chits are being passed, but be pardoned, we'll be having an open session for you all once all these sessions are done. And a gentle reminder once again, this will be live streamed in India as well. So, ladies and gentlemen, let's move to the next session, Dr. Mukesh Chowda. I would like to invite him. He is a senior manager of healthcare services at Solomix Herbaceuticals Limited, India. He is a clinician with a successful track record of over 2 decades. And with his inclination and expertise in research, Dr. Chowda has been the core of research activities in Sri Dhootapapeshwar Limited, which we are immensely thankful, and the Solomix Herbaceutical Limited. So Dr. Mukesh Chowda would present some fascinating insights on evidence-based authenticity, safety, and efficacy of Ayurveda Rasashastra. It's over to you, sir.
Good evening, everyone. First of all, I would like to start my presentation by thanking our Managing Director, Sri Ranjit Pranikji, for giving me an opportunity to present some of our research data. Okay, now this basically, the forum was on Rasa Ushadhis, and Ketan, Dr. Ketan has especially told me that you don't restrict your talk to just Parat Gandak containing products. Okay, you also cover other Bhasma preparations so that we can enlighten the audience what work we have done. Now, basically, as practitioners, okay, majority of practitioners, we rely on
त्याचा संबंध नाही हो. थांबा 2 मिनिटं सर. 2 मिनिटं थांबा. ठीक आहे, तो घ्या. ठीक आहे ना, थांबा सर, 1 मिनिट थांबा. सर त्याचा लॅपटॉपचा नाही आहे संबंध, ह्यांच्या केबलचा इश्यू.
next slide 2 कर, मी इथे थांबतो.
Wait, just wait. काय करताय?
Thank you. So as a practitioners, we always, our basic concern is our medicine should be safe. Whatever we prescribe to our doctors, but our concern should always be that what I am prescribing to my patient, I should know what pharma company is offering me. So this presentation revolves around some examples of authenticity, safety and efficacy in terms of clinical as well as experimental safety and efficacy. And now the era is changing. We are talking of how does the medicine work. So we are also talking of mode of action. Now this is necessary right now because the objective of Ayurveda is broad. Okay, we talk of preventive medicine, we talk of curative medicine, and we also talk of rejuvenative medicines. So we should be able to substantiate it with proofs. Now, when we look at the olden days, what used to happen? when a patient goes to a Vaidya, Vaidya used to diagnose and Vaidya used to prepare the medicines and he used to give and he used to follow up with the patient. Now the era has changed. Right now, when a Vaidya examines a patient, in majority of cases, he relies on ready-made medicines. He relies on ready-made medicines, whatever pharma companies they offer him. And in this process, okay, there are chances of shortcuts. And these shortcuts, ultimately, they lead to reduction in therapeutic deliverables. When we talk of reduction in therapeutic deliverables, it is not just efficacy of the product. It may also lead to toxicity. So we have to be very cautious. And when we talk of safety, when we talk of efficacy, it is joint responsibility. It is not just responsibility of an Ayurveda manufacturer. It is also responsibility of a Vaidya, that whenever I prescribe a medicine to my patient, I should always keep in mind the dose of the medicine, the duration for which I am giving, whether I am giving proper anupan, and lastly, okay, I should never forget that what are the other things like Prakruti, which have to be kept in mind. And keeping this mind, and this is very necessary, because what has happened right now, the Western doctors, okay, they laugh at us. They say that either Ayurveda, here it's mentioned, either it is a placebo or some label it like Dr. Saper. They label it as an toxin. So we have to prove that Ayurveda is not a placebo. Ayurveda medicine, we deliver cure. We are not toxins, provided we are prescribed in proper dose, proper duration, adjuvant and proper diet. So this is the need of the hour. Ultimately, when we talk of research, we have to keep in mind, we have to prove that Ayurveda medicines are effective provided they are used properly. And only effect and no side effect should be our objective. And for this, we require research. I give an example that on the right hand side we see it is an modern insert. Now in modern medicine or in Western medicine, the insert covers more than 50% of the drug information. So we have to graduate ourselves to that level. Sri Dhootapapeshwar Limited, we have already started it. Now when we look at the insert, we can see they have also mentioned drug-drug interaction. They have that ADME profile. We are talking of toxicity. We are talking of dosage in special population, because these are the challenges which an Ayurveda physician what we can say faces daily. Like Madam was giving an example, when I go with a product, okay, they will first ask me, can I use this safely in a pregnant lady? If a person is suffering from HIV, okay, in this HIV patient or in chronic kidney disease, is the medication dosage same or dosage has to be reduced? So all these answers have to be, what we can say, we have to find out the answers. So research is required. So we have to graduate ourselves to the level of Western medicine or even better than that. Now, when we talk of graduation, first thing is standardization. We have to know what is the medicine which we are providing to our patient. Second is safety and efficacy. When we talk of safety and efficacy, it is not just animal safety and efficacy. We talk of clinical safety and efficacy also. And last is probable mode of action. Now, I think in today's our delegate kit, we have inserted one booklet which contains all our research papers, abstracts of our research papers, almost 32 recent research papers we have highlighted. There you can go through it, you can scan the QR code, and you can also download the entire paper which is available on our website. You will understand what kind of work we are doing. And this work was possible through collaborations. And we have collaborated with various institutes in India. And one of our objectives of our Sri Lanka visit was also that we should collaborate with Kelaniya University or University of Colombo, so that more and more researches happens. Now I will give an example of standardization. Now everyone knows Suvarna Bhasma, Suvarna Bhasma, Suvarna Bhasma. Everyone is using Suvarna Bhasma and Suvarna Prashan also. Madam was mentioning Suvarna Malini Vasant, Vasant Kusuma Karras. Everywhere Suvarna Bhasma is, but manufacturing Suvarna Bhasma is not easy. It takes us almost 30 to 35 days for manufacturing Suvarna Bhasma. We have just enlisted the steps. It starts with procuring the gold bars, converting it into a ribbon. We cut the ribbon. Basic shodhan is done in Kanchanar Quad. Then 1:2:3 is the ratio. 1 part of gold, 2 part of mercury, and 3 part of gandak. Okay. First step is mixing 1 part of gold with 2 part of gandak. We amalgamate it, and that golak, okay, it's kept in a sharav which contains 3 part gandak. Now this process, this entire process, when we give this putter, okay, we heat it to almost 750 degrees. And after heating it for 750 degrees, this process is repeated 14 times. And this takes 30 to 35 days, and the end result is Suvarna Bhasma. So we as a company, we know what we are offering. And what we are offering, I give an example in the next slide. This paper is published where the IIT team had procured these samples from the market, 5 Suvarna Bhasma samples. 1 sample was our sample. It was SBB. Now we know what we are offering because we are not following any shortcut. If we look at a SBB, which is our Suvarna Bhasma sample, we don't claim that it is nano. Everything is, whatever is nano doesn't work, okay? Ours was the only sample which contained large size particles to nano size particles. What was important is we have to look at SBC and SPD. Okay, these are also leading manufacturers in India. But if we look at the particle size, somewhere the IIT team, okay, they came to a conclusion that all the particle size are same. If we see particle size are homogeneous, when particle size are homogeneous, it means probably they have not given any Putan Sanskar. It might be procured in a lab or in a furnace. So this is the, all particles cannot be same because when we heat it for 14 times at a temperature of 750 degree, temperature increases, then temperature goes down. It is not possible that we will get an uniform size of gold particles or Suvarna Bhasma. So this SBC and SPD, it's a suspicion that they are not prepared as per Ayurveda text. When they are not prepared as per Ayurveda text, we cannot expect results. So this is what we should know. As practitioners, we should know what I am giving to my patient, what pharma company is offering me. So we have to be very demanding. Second comes toxicity. Now, madam had given examples of Loknath Ras, that she has done acute toxicity, she had done subacute toxicity, she has done subchronic toxicity. What they have not done is 180-day toxicity, which they have planned. Now, for this Suvarna Bhasma, apart from characterization, what we have done chronic toxicity. And chronic toxicity, what is the purpose? We give the medicine, madam had given an example, for 90 days or 180 days in 3 different doses. In their case, they had given 4 different doses. One was therapeutic dose, 5 times the therapeutic dose, 10 times the therapeutic dose. Ultimately, the objective is, what is the non-toxic level? It is called as NOAEL, no observed adverse event level. Now this, for our Suvarna Bhasma, was 333.33 milligram. Because as per classical text, the dosage or matra of Suvarna Bhasma is a custom Rati. We had taken the double dose. A custom is 15 milligram. We had taken a dose of 30 milligram. So 30 milligram into 10 times, so almost more than 300 milligram per day, it was safe. So what does it indicate? That whenever you use Suvarna Bhasma, please do not exceed a daily dose of 333 milligram. Now there are some more examples which I am giving. One is Rasashastra Ras, and second is Swaskas Chintamani Ras. Now why these examples? These are our recent publications. Now these medications, Rasaraj Ras, whenever it is used to treat chronic Vata Vyadhi, or Swaskas Chintamani, when we try to treat lung fibrosis or tamakshwas, it is not given for 2 days, 5 days or 10 days. It is used for a longer duration of time. So chronic toxicity, this is a basic prerequisite which we should demand. And this we have done. These are our recent 2022-23 publication. And one of the most important thing, madam, for Loknath Ras also you have planned. It is genotoxicity and mutagenicity. Now, for Suvarna Bhasma, we have already published it. Now, I would like to just zoom this slides. Madam, see, this is vehicle control, and next to it is Suvarna Bhasma. We can see intact chromosomes. we can see intact chromosomes. It is basically a chromosome aberration test. But when a known genotoxic agent, which is mitomycin C, was used, we can easily see there are damaged chromosomes. So this kind of work has to be done for all the bhasmas which are widely used. So this is proven, this has already been published. And there is one more test, which Madam had mentioned. Madam, you spoke of comet assay. Madam, we have done it for our Suvarna Bhasma, and we have done it for the therapeutic dose and 10 times the therapeutic dose. Now, basically, what is the principle of comet assay? We separate lymphocytes, and in these lymphocytes, we look for DNA fragmentation. If there is DNA damage or DNA fragmentation, we can see this image, which looks like an comet. Here, cyclophosphamide is a known genotoxic agent, which is documented. So we can see this comet-like appearance. But this comet-like appearance was not seen in Suvarna Bhasma treated groups. So it proves that Suvarna Bhasma manufactured by Sri Dhootapapeshwar Limited, okay, it is not genotoxic. Secondly, Ames assay. Ames assay is the gold standard to rule out whether any chemical entity has that cancer-causing potential. Because in Ayurveda, what happens? It's one of the oldest science. We say health system of medicine. We follow the model of reverse pharmacology. We go from men to mice model. Okay, but in allopathy, they follow mice to men model. So Ames test is very important. If any new chemical entity fails the AMES test or Ames test, I would call, the dossier is closed. That product is never launched because it is confirmed that this is going to cause cancer. So this test also, so it is very unfortunate that for our old products, like Ayurvedic products, now the regulators are demanding this type of data, genotoxicity or mutagenicity, when these medicines have been used for a very longer duration of time. So this is very important. Second is, see, although our director said that you have to cut short your presentation, I might take 5 to 7 minutes extra. So we have proved that it is not genotoxic, it is not mutagenic. And one more thing, it's not just, we have to use some innovative models. Like for Suvarna Bhasma, we had used zebrafish model also. In zebrafish, I would like to just play this video. The left side, we can see that zebrafish, they are swimming at the base of the tank. And on the right-hand side, it was Suvarna Bhasma treated zebrafish. Now, they preferred to swim in the upper zone. Now, what does it indicate? It is a sign of non-neurotoxicity. When we talk of, they are very, what we can say, protective. This anxiolytic effect, or made the effect of Suvarna Bhasma, okay, increases their risk-taking behavior. So they prefer swimming in the upper zone. Now this has been, what we can say, depicted in the form of graphs also. If we see these graphs, this is the vehicle control, Suvarna Bhasma. We have used comparative control. Now this comparative control is alcohol. Now alcohol is better than Suvarna Bhasma, but it is not a medicine, no? We know alcohol is an anxiolytic, but we had purposely used it. At the same time, we had used caffeine also. Okay, now caffeine is a suppressant. So this fish preferred the lower dose. So this proves that our medicines, okay, these are non-neurotoxic. And if we look at the next graph also, we see on the top, we see that alcohol-treated group, then come this Suvarna Bhasma. Now this graph depicts the height at which they are swimming from the base of the tank. So this proves that these are Suvarna Bhasma is non-neurotoxic. And now when we talk of anxiolytic effect, nowadays everyone knows stress. When cortisol level increases, when stress increases, we are more prone to infections like upper respiratory tract infection. Immunity reduces. So this is one of the benefit of reverse pharmacology approach, where we can interpret the results of these findings. Yesterday we were, Dr. Bhushan Patwardhan sir was talking of rejuvenative potential also. Now this is a classical example. This is Vasant Kusumakar Ras. Okay, Vasant Kusumakar Ras, has that tissue protective effect. We say it has an protective effect on pancreas. Now, when we talk of Vasant Kusumakar, it is not the medicine to reduce blood sugar levels. No, it is not our objective, because I think Yogesh in his presentation he told that basically the culprit is chronic hyperglycemia. This chronic hyperglycemia leads to end organ damage. When we talk of pancreas, okay, if blood sugar levels are under control, and we are talking of insulin levels also, because when we look into Asian scenario, we have more people who are obese. We are facing an epidemic of diabetes. Obesity combined with diabetes. So first thing which we have to tackle is insulin resistance. So that insulin resistance is also important, which is very important in tackling the diabetic epidemic. Now we talk of HbA1c level reduction with Vasant Kusumakar Ras. We talk of plasma insulin reduction with Vasant Kusumakar Ras. But ultimately, what is that effect? I would like to show the next slide. This is the immunohistochemistry slide. Okay, in this immunohistochemistry slide, if you look at the center, this is basically a pancreatic section and islet. It is an islet. If we see, you can see some brown dots. Now these brown dots is basically immunohistochemistry. It is SIRT1 expression, silent information regulators. Okay, this SIRT1 expression is very important because reduction in SIRT1 expression is an indication of organ damage or fibrosis in the pancreas. Now this, when we talk of diabetic control group, where no treatment was given, there is reduction in SIRT1 expression. When we talk of Vasant Kusumakar Ras, that brown dots are very prominent, which are almost similar to the vehicle control group. It means our medicines have that Rasayana effect, and we have proven it through this studies, and that further studies have been done by Dr. Yogesh. I would like to cite one more example. Neuropathy he has already presented. We say that Vasant Kusumakar Ras reduces the progression of diabetic neuropathy. How? By inhibiting the WNT beta-catenin pathway. Now, right now, US FDA has recommended the use of pregabalin or gabapentin. Now, if you look at the article which we have cited, that this is a review article. It is mentioned, it's in BMC, Biomed Central, okay. It says, it is 2021 review article, that pregabalin increases the risk of myocardial infarction. Pregabalin increases the risk of deep vein thrombosis. Pregabalin increases the risk of LVH. Now this, whatever adverse events we are mentioning, these are also the adverse events of diabetes. Now when we talk of pregabalin doing this, then no medicine is safe. And Ayurveda has been questioned. But when we talk of Vasant Kusumakar, it has that efficacy which is comparable to pregabalin. But what is the benefit? Sir, pregabalin is not anti-hyperglycemic. Pregabalin is not pancreatoprotective, okay. It is neuroprotective. It is especially used for pain management in diabetic neuropathy. So Ayurveda definitely has edge in this segment. When we talk of our Asian population, first comes pre-diabetes, then comes diabetes. So when we talk of pre-diabetes, it is basically insulin resistance. So we have one more publication where we have studied our formulation Madhumaya Kusumakar Ras, that it what we can say, reduces or reverses insulin resistance. Now, why insulin resistance is important? Because when we talk of diabetes, or whether we talk of NFLD, or whether we talk of polycystic ovary syndrome, insulin resistance is present everywhere. And one of the major thing is metabolic syndrome. What is metabolic syndrome? Is 5 things. No, one is obesity, high sugar, high blood pressure, high triglycerides, and low HDL levels. Now to tackle this, we have to reduce HOMA-IR, we have to reduce insulin levels, we have to reduce triglyceride levels, and this formulation has proven that, okay, it works there. And here, if you see, we have taken a allopathy medicine as a comparator, metformin. Metformin does not reduce triglyceride levels. We can see this in the graph. Metformin does not increase the, reduce the LDL levels. Here, Madhumaya Kusumakar Ras or an Ayurveda intervention was better. So we have to, what we can say, think of Ayurveda. What we can say, whenever a patient with pre-diabetes comes to us, no need of giving metformin. lifestyle modification along with Ayurveda interventions like Madhumeh Kusumakar Ras, patient will definitely improve in terms of what we can say pancreatic protection also. One more example which I would give would be Rudrok Chintamani Ras. Now this Rudrok Chintamani Ras, we are talking of preventive cardiology. No? We are not talking of, we are not competing with allopathy medicines for heart. If I have an heart attack today, take me to an Ayurvedic doctor, or you will not treat me, madam, you will not treat me. Okay, I have to go to some casualty. Okay, now what happens, then we should know our limitation. We should know our limitation. Okay, in acute condition, it might be better, but when we talk of preventive cardiology, Ayurveda definitely works. Bruhatva Chintamani definitely works. Now here also we have seen SIRT1 expression. See, in the Vasant Kusmakar Ras slides, there was SIRT1 expression in the pancreatic tissue. This is SIRT1 expression in the serum levels. And secondly, what we had done, CTN levels, cardiac troponins. So whenever there is myocardial tissue damage, there is increase in cardiac troponin. So here also we had artificially induced an heart attack by injecting isoproterenol. So TROP-T increase, SIRT1 expression had reduced. There was improvement in SIRT1 expression and reduction in CTN levels. Secondly, chronic fibrosis, what does it lead to? Heart tissue damage. There is cardiac hypertrophy. Now this is a special staining. It is called as trichrome staining. You can see 4 quadrants. First upper one is vehicle control. It is normal myocardium. Next to it we see a blue part. It is basically a special trichrome scanning. It will only stain the fibrous part. That blue part is entirely fibrosis. It is disease control. Now, when we talk of Rudrok Chintamani Ras or Bruhatva Chintamani Ras, there is reduction in severity of cardiac fibrosis, so less myocardial damage. Now, this is very important because I had attended one lecture of a cardiologist. His last slide was this. This is a famous, what we can say, heart transplant surgeon, Christian Bernard. He said that I have saved the lives of around 150 people by heart transplant. If I had focused on preventive medicine earlier, I might have saved 150 million lives. This applies to Ayurveda. No, this is what is Rasayan therapy. So we are talking of preventive cardiology. Then comes, now Suvarna Bhasma is over. Now we are also talking of Rasa Sindur and Kajjali. Probably it is almost present in all our formulations. So first of all, we have to clear our queries that our Kajjali or our HGS is different than mercury chloride, HgCl2 or mercurous chloride, Hg2C, it is different. Now this work was independently done by Indian Institute of Technology, Dr. Jayesh Bellar and team. We had not funded this project. They had procured our samples from the market, and what they found that, okay, HgCl2 increases the cortisol levels, but HGS, Rasa Sindur and Kajjali, they do not increase the cortisol levels. Now, most important thing was HGS. This HGS was lab-made HGS. So when we talk of HGS is not toxic, HgCl2 is toxic. Now we cannot compare HGS with our Rasa Sindur or Kajjali because in their 2022 paper, they have clearly mentioned that HGS, Kajjali and Rasa Sindur are different. HGS is 50% mercury, 50% sulfur. But when we talk of Kajjali, it is alpha HGS and Rasa Sindur is beta HGS. Now these references are in chemistry textbook. This is a very beautiful paper. And secondly, on the right hand side, if you see that graph, HgCl2, there is mercury deposition in the zebrafish tissue. But when we talk of HGS or when we talk of Rasa Sindur or when we talk of Kajjali, there is no deposition. There is no deposition. It does not mean that we can replace HGS with Suvarna Bhasma, no, with Kajjali or Rasa Sindur. No, it might, we are talking of safety. It is not toxic, but therapeutically it may not be effective, no, which we are claiming for Rasa Sindur and HGS. And one more thing which I would like to highlight is we had done previously 10 years ago work with our regional research lab Jammu, where we could prove that Rasasindur improve the bioavailability of rifampicin. Rifampicin is a very well-known anti-TB drug. It is very toxic, but it is poorly bioavailable. So we have to give it in a higher dose. So in our study, we had proven that it increases, Rasasindur increases the bioavailability of rifampicin by almost 40 to 42%. So this is great, but unfortunately, we cannot launch this product. What will be it? It cannot be called an Ayurvedic product, and it cannot be called an allopathy product also. But our products are effective. We say, no, in every formulation, Rasa Aushadhi, we have Kajjali, we have Rasa Sindur. It has its own therapeutic effect, but they also work like a catalyst also. No, they improve the bioavailability of the accompanying drugs. Now, this is a slide many people ask, does Ayurveda work on hormonal levels? Yes, definitely Ayurveda products, they work on hormonal levels. Madam had given an example of Sri Vyadhi Hariras. See, these are the two graphs. I have not mentioned the products. On the left-hand side, when we talk of 17 beta estradiol induced oligospermia, there is reduction in testosterone levels, and our Bij Pushti Ras improves the testosterone levels. On the next hand side, we talk of letrozole-induced polycystic ovary syndrome. It is basically hyperandrogenemia. There is increase in testosterone levels, and when we talk of polycystic ovary syndrome, Madam had mentioned in her slide, there was ovulatory dysfunction. Now this ovulatory dysfunction is because of reversal in LH-FSH ratio. Usually it goes hand into hand. So this reversal of LH-FSH ratio is possible through Ayurveda medicines, and we have proven it. Now, after this study, we have taken it up for clinical studies, because ultimately what will help Ayurveda is evidence and documentation. Now, these are some of the examples of other bhasmas. Like for Asti Poshak, this paper was published last year. Our principal investigator was Dr. Pawan Godatwar. It was our first project with him, which we had done in NIA Jaipur. Now, what happens? What is the edge of Ayurveda calcium over allopathy calcium? See, allopathy calcium is calcium. Allopathy calcium, to maintain or improve calcium deficiency states, it is fine. But this allopathy calcium will not work in senile osteoporosis, will not work in postmenopausal osteoporosis, because their pathophysiology is different, no? Reduction in estrogen levels reduces the bone mass. So where we have to give some medicines which have phytoestrogenic action. Modern calcium will only improve the bone mass, but it will not stop bone resorption. Now, through this publication, we have proven that our product, an Ayurveda intervention, Asti Poshak tablets, improves bone mineral density. At the same time, it also stops bone resorption. Now, specific markers are used, bone-specific alkaline phosphatase, serum osteocalcin level. These are some of the latest markers which are used. So we are also spending a research. Why? Because to help the mankind. Otherwise, you doctors will not believe us. Means a tradition of 150 years, or when we talk of Ayurveda, 5,000 years old, now this story will not sell, no. People are talking of evidence, so we have to generate evidence. Next is Amla Pitta Mishran. So nobody was ready to take this study, because we had published one experimental study in Ancient Science of Life, followed by, after 10 years, in 2022, we had published a paper in GIM, where an endoscopist, where an Western gastroenterologist, he endorsed that your product works. After giving it for 1 month, there was a reduction in endoscopic scores in terms of stomach inflammation. At the same time, what we were discussing, it's not just endoscopic changes. When we talk of science, we have to use Ayurveda parameters. So in this study, we had used Amla Pitta symptom rating scale score. Now this classical symptoms of Amla Pitta, avipaka klamotklesha tiktam lodgara gaurave, rut kantadaha arochebesha Amla Pittam vadet bishak. Now these are seven symptoms. Based on the severity scale of 3, maximum score was 21, and we saw that in addition to changes in endoscopic score, there was symptomatic improvement also. So, and this findings we have confirmed through a data of 204 patients. I would quickly conclude. Yesterday, Dr. Bhushan Patwardhan was talking. Why does a product work? Product works because it has a strong pharmacopoeial rationale. When we talk of Amla Pitta Yoga Ratnakar, we talk of Pachanam Tikta Bahalam Pathyam Cha Parikalpayet. This is the Chikitsa Sutra. Now, this Amla Pitta formulation containing Shaktik Bhasma, Bhunimbadikvat, fills perfectly, justifies this Chikitsa Sutra. Secondly, it is basically revalidation of Amla Pitta symptom rating scale score versus what we call today is functional dyspepsia in Western medicine. Then Ayurveda can also play a positive role in IDA management. We have reconfirmed it. Yesterday again, Dr. Bhushan Patwardhan was talking, whatever small studies we do in Ayurveda, we cannot replicate, but we have replicated 2 studies. One was for Amla Pitta symptom, for Amla Pitta, second was for Abralo also. In our phase 4 study, we have proved that our medication, Abraloho, okay, guarantees and HB rise of 2 gram per deciliter in 8 weeks time. Lastly, the second last slide, we were talking of immunity. So we are the first company who have published a paper, and we have not just talked of quality of life, we talked of specific parameters like increase in CD4 count. Now the CD4 lymphocytes are the ultimate cells. Reduction in CD4 cells will lead to opportunistic infection. So we have proven that, okay, our medicines like Swamla, Chamanpras, they have the potential to improve the CD4 count. Thank you.
Thank you very much, sir, Dr. Mukesh Chowda, for that evidence-based authenticity, safety, and efficacy on Rasashastra. So I would like to invite Dr. K.P.K.R. Karunagoda, the senior lecturer and head of the Department of Shalya-Shalakya, Prasuti Tantra and Kaumara Brutya of the Faculty of Indigenous Medicine, University of Colombo, to present the token of appreciation. It was a great presentation presented by Dr. Mukesh Chowda. As we all are aware, Dirgha Mayu Smruti Meda Arogyam Paramam Vayam Prabhavarna Shotodara Dehendre Balamparam Vag Pravritti Pulatikanti Labatena Rasayanam. So ladies and gentlemen, we talk about these benefits of Rasashastra, and we need to have the concluded safety, efficacy, authenticity and standardized parameters. So Sri Dhootapapeshwar Limited has successfully concluded from the time tested to evidence-based medicine. Yes, they have done through their research and immense development, coordinating with the relevant institutions and so on and so forth. As the time is limited, let's open up the forum for the open session. And 3 questions, right? Okay, so the first one I do have with me. Please explain the Rasashastra as it can be used in pediatrics. I think it's posed to Madam Acharya from a personal cheat. Yes, one more. Can we have a microphone, please?
I have mentioned before, Bala, it is very important from Swarna Prashana. In Kashyapa Samhita, it says immediately after birth, Swarna Prashana is mentioned. I try to follow this in my center. Immediately after birth, if the area, everything is fine, I tell the father to put a drop of Swarna Prashana to the baby immediately after birth. Then I follow 6 months of Swarna Prashana, not that Pusha Nakshatra one. I tell them to start on Pusha Nakshatra and go for 6 months. This is also mentioned in Kashyapa Samhita. And many conditions of autism and ADHD and different conditions, we have beautiful medications, especially from Dhootapapeshwar Limited. I give this Swarna Mahayoga and Brahmi Yukta Swarna Yoga. That has proved. And one patient of mine, you must have heard of Proseria. Proseria is like that baby is born and then immediately they brought to me. She couldn't come for this Garbhini Charya and before conception also because of the COVID. After that, because they both, the husband and wife, had some health problem. So I was expecting this. I told them to undergo this Garbha Sanskar. They didn't go and they had a Proseria baby. And first one year he was not doing anything. Then with this Mahaswarna Yoga with Brahmi, he's responding to people. Last week also he had come to my clinic. He's trying to talk to people now. He's a 2-year-old now. So we have many Rasashadis, we can give during this for children, especially in immunity. Even their Shishubharana Rasa I regularly give for a month or two because in Bangalore mostly children fall sick with cold, cough because of the pollution and cold weather. So Swarna Bhasma is the best for children. I hope it will.
Yes, thank you very much, madam. And moving to the next question. 20-year-old patient, diseases, mitral valve prolapse and murmur. Can it be treated with Roga Chintamani Rasa or Makaradvaja?
Sir,
yes, a 20-year-old patient, this is mitral valve prolapse and murmur. Can it be treated with Roga Chintamani Rasa or Makaradvaja?
Sir, I would like to answer this question. If it is an anatomical problem, you understand, okay? If
yes.
It is an anatomical problem, he might require correction supported by Bruhatvan Chintamani Ras, because then it should not be a very broad claim. If it is an anatomical problem, we have to fix it. Okay, means I have in my practice, I have not heard treating or correcting mitral valve. There might be
yes.
good doctors who might be doing it, but I have not heard it. If you ask my opinion, okay, if he requires correction followed by Rasayan therapy with Bruhatvan Chintamani Ras can definitely help.
If there's an anatomical correction, it need to be done. Right, yes. And proceeding to the final question to Dr. Yogesh Kulkarni, can you please explain the VKR in acceptable dose will reduce proteinuria and GFR in CKD stage 1?
No, as far as clinical dose of VKR is concerned, obviously it has shown some good effects in kidney. But again, we have to prove it scientifically by using the same doses, whether it can be useful in CKD or not. So I think a basic study is required for its effectiveness in CKD. Yeah.
See, we are talking of treating CKD, or
yes.
we are talking of preventing CKD, because when we talk of CKD prevention, okay, the mode of action by which an NaCID damages the kidney, and by the way the hyperglycemic damages the kidney, okay, it is different. When we are talking of, these are two different mode of action. When we are talking of preventive diabetes related chronic kidney disease, Vasant Kusumakar definitely works, and we can get n number of examples. Our Vaidyas are using it, but they are not documenting it. So this is definitely possible.
Yes. Right, okay. Thank you very much for those valuable insights. We need to wrap it up, this open forum, as we are running short of time. And next, I would like to invite for a short address Dr. S.L. Vevalvala, the Senior Lecturer at the Head of Department of Ayurveda Basic Principles of Faculty of Indigenous Medicine, Kampaha Vikramarachi, University of Indigenous Medicine.
Yeah, right, okay. Good evening. Yes, I'll take only 5 minutes, right? All the dignities, madams and colleagues and students, right? This will not be a speech, right? I need to draw attention, and I need to make a humble request from all the dignities and the policymakers and from the academia. I'm from the section of philosophy, part of philosophy. This is what the philosophers are doing, right? We think differently and suggesting, right? We need a huge paradigm shift about how we are thinking. Before a couple of weeks, I had a chance to, yes, I was developing a syllabus for Bachelor of Education with Professor Pujitha Wickramasinghe, who is a renowned pediatrician from University of Colombo, Faculty of Medicine. He asked me 2 questions, right? Why your students are demanding to teach pharmacology, right? And second question was how your drugs are acting. Believe me, his jaws were dropped with my answer. So today we witness very precious and great findings, right? That kind of research are very need, sir and madam. We need this kind of research about uh, standardization, safety, and efficacy. It's right. But, Madam Priyani, yes, I got this idea with a word said by Madam Priyani, is that super qualities, right? We need super way of mechanism to explain the super qualities of our drugs. That's it. And in your speech, Dr. Mukesh's speech, you had some trouble of explaining our calcium and allopathic calcium. There's a difference anyway, no? But we can explain why. This is the reason, right? We are struggling with the level of molecular biology. Our thoughts has been end up with the molecular biology. But our philosophy and all our theories are based on molecular physics. I agree. This is my humble request from our authorities and our academics. It is time to acquire, or it is time to teach our students with the molecular physics and biophysics. Right? We are dealing with physics apart from the, yes, actually we are not offense with the chemistry, right? But if you take any single phenomena, it could be explained, right, in the level of chemistry, and but also with the level of physics. Laws of physics are ruling this universe, right? There's each and everywhere we see physics, right? If we need to explain our theories, which cannot be explained through molecular biology or modern pharmacology or molecular chemistry, the reason is they are acting under the laws of physics. So if we are trying to amalgamate this knowledge into our stream, right, it will be very easy to explain our things, especially our theory of filaments, Panchabhutavada. Personally, I believe, as a philosopher, as a man from philosophy area, right, we should amalgamate that knowledge into our curriculum. And why I took this opportunity in this Rasashastra Sambhasha, because this is about rasa. Rasa dravyas are acting very differently. If you go and see Sushuta Sanghita, Sutrasthana 46, right, it clearly mentions a term ashukari. It defines that molecules are acting in a certain place, accepting other dhatus, right? It acts directly to a certain dhatu, but we cannot explain it using the conventional methods, right? This is not time. I can tell you some other examples, but this is not the time. Another small example, right? In Sushruta, it has explained the water molecule, right? Water is acting two-way pattern, right? As Ayurveda physician, we use water, hot water for some diseases and cold water for some diseases. Before a couple of years, Faculty of Medicine conducted research on diabetes mellitus, and they told that if you are using hot water, your progress is good. But they couldn't explain it, but we can. Why? It works according to the laws of physics. So, dear Madam and Sir and authorities, and I suggest this type of companies, they are doing a very huge thing and doing great research and supporting to our education system also. So please do conduct research with the collaboration of physicists. Please combine the theories of physics into Ayurveda, then it will be very easy to explain, right? I have gone through some recent researches. Now India is doing such things. So I invite the deans, Kamal sir, and our other academia, please amalgamate those knowledge into our VMS students. And I suggest this kind of companies to conduct or allocate some kind amount of money to this kind of researchers, right? Thank you, sir. Thank you very much.
Thank you very much, sir. And proceeding forward, the latter end of this program, I would like to invite Professor S.G. Ranasinghe, the chair of this esteemed Rasashastra Sambhasha, to address the gathering.
Members of the organizing committee, Commissioner of Ayurveda, my colleagues as co-chair at the head of the table, honored participants and delegates from India, friends and ladies and gentlemen. I have the pleasure of speaking to you as the chief guest of this symposia, which I consider as a privilege and honor bestowed upon me. And also I want to thank the committee members who organized this symposium for giving me opportunity to address you in few words. Now we have arrived at the terminal stages of this seminar, and I don't want to take much of your time, but I would be brief and to the point. I am aware that this symposium is entirely devoted for Rasashastra, and this is the 6th one. The previous one was held in 2019 at the same venue, but due to certain conditions prevailed in this country, we have to wait for 2 or 3 years to have this present symposium materialized. And I thank the organizers and Sri Dhootapapeshwar Limited India for take all the efforts and all the occasion to have this seminar a success. While I attend the seminar from its beginning, I was compelled to observe that this is a special occasion for Ayurveda, especially to Rasashastra. I have observed that the committee members and the organizers have taken all the efforts to surmount the difficulties and obstacles prevailing here. Now the condition is a little bit congenial for this type of academic work, and they have taken all the opportunity to materialize this symposium. Charaka and Susruta emphasize the importance of Sambhasha, that is symposia, where we are going through the treatises of Charaka and Susruta. We have quite evidence that even at that time, this type of Sambhasha and symposia were took place. Rishis as gurus and their disciples as students have this sort of symposium through a way of discussion. And they have emphasized the benefits we can reap from this type of exchange of views, and we can present our views from our research work, and also we can alleviate some problems coming to the particular discipline by having necessary Sambhasha at equal intervals. But I should say that organizing a Sambhasha of this nature is a formidable task by itself. Sambhasha means a collective effort of all the resources, and these resources are of variegated nature. We have to collect the human resources, and we have to get used of material resources, and you have to utilize financial resources also. Those are the certain very important resources we have to touch upon if we have to have a successful symposium like this. Now, when I follow the discussions, and I wanted to express myself, so I was wondering what message I should give to this august audience after going through the entire research work presented by these learned professors and presenters. If I say the advantages of Rasashastra, that would be a repetition, because all know what are the advantages of Rasashastra, and I don't want to repeat that. And if I say all the praiseworthy sayings, it would be a hypocrisy. So what I thought was, I want to sort of analyze the presentations done here in a logical way. And I have noted certain highlights where these research workers has present to us. And these highlights I want to pinpoint, but I don't want to elaborate on this because of the constraints of time. And I just want to mention how far we have matured in our research activities. First, it is the extent and depth our research workers have achieved in surpassing the research objectives. So that is a very pertinent question in research, where objectives has to be first laid down, and all your research work should be undergoing to achieve that objectives in a positive way. That itself shows how well versed with these subjects by the presenters today, and that is commendable. Second, I have noticed the way they interpret their observations and results by using other disciplines also. So it would become a multidisciplinary seminar at this point. And I noticed that most of the workers stress more on modern science and modern medicinal literature. That's also a very good approach, but at the same time, I should say that Ayurvedic philosophy and Ayurvedic principles, especially with regard to Rasashastra, should be dealt with. Thirdly, I am actually surprised to the dexterity and the suppleness they harnessed the modern research methodology into their research work. Now, modern research methodology is entirely a brainchild of the West. But Rasashastra is of Oriental nature. So you have to couple with these two in order to bring fruitful results. The way these research workers done that is actually surprisingly good. Then other highlight I noticed is how they try to generalize their findings. Now that is very important. The modern science developed through generalizations. Now for example, we take a project for our research and that's a unit entity. We are using a certain type of disease and we are using certain type of drug. Here, Rasa Aushad. Then that unit investigation has to be extrapolated to generalize. That's how the modern science developed. Experiments and research work, they want to collect this data and they did actually, they generalized those observations and inferences. Now inferences are two types, these factual inferences and statistical inferences. Both these could be utilized in informing generalizations. What I gathered from here is that generalization was not very enough, but that is excusable because all these research works undergone and presented here are in the nature of pilot studies. So to generalize, you have to go further, and in further stages, you have to collect more data, and you always try to connect with, couple with your basic principles and try to extrapolate into formation of generalization. For example, now I give you a simple example. Now from ancient rishis, they have observed certain things connected with the environment, and some patients come with headache, some with swelling of legs, and they always try to correlate with the symptoms with some sort of philosophy. Now that is the observation. That is also a research experiment. But ultimately, after gathering so many hundreds and hundreds of observations, they have come to the conclusion in a generalized way that how this Vata, Pitta and Kapha theory evolved. So that's a generalization. In modern science also the same thing happens. Modern science took leaps and bounds today as an advanced science because they were very successful in generalizations. Now all these theories and laws and all these things are generalized conditions. They come out of individual research. But here I want to stress that in time to come we will follow the same regimen. So as soon as we got our results and observations and inferences, try to generalize them. Then only we can develop our own theses, then only we can develop our own methodology with regard to this context. And the other thing is, I have noticed that our research has actually attained the maturity. This is, though we are concerning about the pilot studies, that pilot studies are very well advanced, and you have taken all the help from the allopathic medicine, histopathological studies, and all these serological observations. That is very good. But at the same time, we have to take in mind that this is a separate science, originated from thousands and thousands ago as a separate entity of medicine. Now, going beyond certain parameters and relying much on modern medical paraphernalia, it is good and it is welcome for the argument purpose, but as a science, we have to develop our own entities. So that's why I am always stressed on the philosophy of research. What is the philosophy of research in Ayurveda? I don't see, even in India or Sri Lanka, we have thought about it. Now we have so many institutions doing research. BMARI at Navina, then some institutes affiliated to universities, they do very good research. But I want to know what is the philosophy behind Ayurvedic research. Are they going to invent new drugs? Or are they want to establish scientifically already drugs we are using? Or are they trying to dislodge Western medication by improving our research and relying on Western medicine parameters? Are we hope to dislodge already established Western medicine, which would be a very difficult situation here, because we all know that that all the Western drugs have toxic effects and side effects. But still, having all these handicaps, Western medicine is riding high. So actually we have to think about this, because this is the proper time to think about this. What is the philosophy of our research work? Are we doing meaningful research, or are we just taking FSR projects, and are we using the same methodology where Western medicine people are doing? So that is very important in the sense of future work. Now with these words, I wish all the success to Sri Dhootapapeshwar Limited for their future endeavors, and also I have to give this message also. It's always good and always better and always convincing if we rely on Western paraphernalia, Western parameters, well and good. But at the same, don't forget that this is an entity of its own. Relying too much on other subjects will automatically and without even knowledge to us will hinder our originality of our medicine. Now with this comment, I want to wind up my chief guest delegation delivery. But again, I am very much thankful for me to give the opportunity to speak few words to you. And I hope in time to come, you will continue this effort furthermore. And we are always very keen to participate and express our views in future also. Thank you very much for your attention.
Thank you very much, sir. Professor S.J. Ranasinghe, the chair of Rasashastra Sambhasha Edition 6. So I would like to invite Mr. Ranjit Puranik, the chairman and managing director of Sri Dhootapapeshwar Limited, to present the token of appreciation to Professor S.J. Ranasinghe. And at the same time, I would like to invite Dr. K.P.K.R. Karunagoda, the co-chair and the senior lecturer and head of Department of Shalya-Shalakya, Prasuti-Tantra Kaumara-Prutya at Faculty of Indigenous Medicine, University of Colombo, to receive the token of appreciation. and also the co-chair, Dr. S.L. Vevalvala, the senior lecturer and head of Department of Ayurveda Basic Principles of Faculty of Indigenous Medicine, Gampaha Vikramarachi University of Indigenous Medicine. While remembering the immense contribution of Dr. Dhammika Abhay-Gunawardena, the Commissioner General of Ayurveda, I would like to please Dr. Heva Gamage, the Assistant Commissioner, Technical Department of Ayurveda. Sir, I would like to invite to get the token of appreciation from Mr. the Chairman and Managing Director of Sri Dhootapapeshwar Limited. Also, the Dean of the Faculty of Indigenous Medicine, Sir, Colombo, and Faculty of Indigenous Medicine, Gampaha Vikramarachi University of Indigenous Medicine, being the knowledge partners for the organization from Rasashastra Sambhasha-6, Sri Dhootapapeshwar Limited. Inviting the Commissioners of Ayurveda, Dr. K.M.P.S. Sirivardhana, Western Province, Dr. A.D. Kumarage, Sabaragama Province, Dr. Sridhar, Eastern Province, Dr. J. Ganeshan, Northern Province, Dr. J. Herath, Central Province, Dr. P.G.D. Prematila, the Commissioner of Uva Province, Dr. C.L. Guruge, the former Commissioner of Southern Province, Dr. Dhanani Senasekara, the former Technical Commissioner of Ayurveda, and Dr. Priyankini Godage, the Commissioner of North Central Province. Thank you very much, respected dignitaries, for being present here at the 6th edition of Rasashastra Sambhasha, organized by Sri Dhootapapeshwar Limited. As we all know, the authenticity, the standardization, the safety, efficacy, all the parameters when preparing a rasa drug, we consider about many. The safety, the histopathological safety, the biochemical safety, the non-neurotoxicity, the non-mutagenicity, the non-genotoxicity, many more considered by the manufacturing process of Dhootapapeshwar Limited. Thank you very much for being here over a decade, over 1.5 decade in Sri Lanka. And at the same time, to propose the vote of thanks, I would like to invite Mr. Ranjit Puranik, the Chairman and Managing Director of Sri Dhootapapeshwar Limited.
It's very strange when a vote of thanks first starts with an apology for not being able to keep the time. So I do apologize on behalf of us as organizers for not really keeping the time, and I think it's prolonged. It is automatically a kind of fatigue that sets in. But nonetheless, I think the retreat was complete. Chairman sir, your words have been carefully listened to. Chairman sir, very briefly, but I think you have spoken very deeply. That we in the Ayurveda sector, it doesn't the philosophy of our research. And how do we place it? What is going to be forward? Sure that we realize this potential from our ancient medical heritage. And I think this is a point very big to ponder. The unfortunate part, I think, so you will agree, is that we will only know if we are right after the event has passed. And the good part is that we can continue to correct ourselves and do this course correction as long as we keep in mind that are we really achieving our full potential with regards the inheritance of this great medical science that we have. The Rasashastra Sambhasha was formed with a very simple philosophy, and it's given right up here on the board. It's an Indo-Sri Lankan forum for exchange of views and sharing of experiences on the science and practice of Rasashastra. To a very large extent, this forum also came about not because we are a manufacturing company, not only because we manufacture products and wish to sell more products, but also as a confidence boost within the sector, and a confidence boost from talking about minerals being purposefully intentioned in formulations with a therapeutic goal and not really to be classified as heavy metals. That stigma was something that we did not like. We believed that much more science needs to be brought in and much more science needs to be portrayed. And that's the reason this forum came about. I think what we saw today was the sixth edition of this forum that brought about this need for a multidisciplinary approach. And I think that is something that's come about. I think all of you will be quite pleased to note that a very premier institute like the IIT or the Indian Institute of Technology is today accepting PhD scholars who from even the Ayurveda stream. So they're ready to take on PhD scholars, and the PhD scholar would do it at behest of a IIT anywhere in India. So as I understand, this multidisciplinary welcoming of Ayurveda graduates, of Ayurveda scholars, is very much there. I do believe that this multidisciplinary approach that is being followed today was an example of that. We also had learned practitioners talk to us about their own experience with Rasa products, and I think this is the kind of the speed that we require. I will now turn to the task at hand, which is the delivery of the vote of thanks, and let me start by thanking Government of Sri Lanka, especially Honorable Minister Sisir Jayakodi ji. He has been extremely cooperative. I have, over a period of the last year, met him thrice in India, and he has taken the trouble to travel to India. He has taken the trouble to even be here on both the days of this entire IUREX, and it is with his blessings that not only could we revive Rasashastra Sambhashan and take it to this restart mode that is there. Also thank Commissioner General Dr. Dhamika Abhay Vardhana, because he has also been like an elder brother, encouraging us to ensure that there is a lot more collaboration between India and Sri Lanka, a lot more science that is being discussed, a lot more exchange of students and experience that is exchanged. I would like to thanking both of them first for this entire forum. And you know, today we had the this learned Professor S.G. Ranasinghe, who chaired this entire meeting, and I think his brief 6 minutes tells us about the kind of insight and the kind of thoughtfulness that we all need to take home from his message. I thank you, sir, for your patience, and I thank you for sparing your time and coming across and chairing this entire meeting. And I would also take this opportunity to thank both our co-chairs, Dr. KR Karunagoda, Madam, thank you for attending this entire session and being our co-chair, agreeing to be our co-chair. And of course, the second co-chair that we had was Dr. S.L. Vevala. Sir, your thoughts about dedicating more resources towards research, dedicating more projects, dedicating a very firm program that is continually available to students and scholars. That point is noted, that point is taken on board, and hopefully in our discussions with University of Kelaniya, as well as some brief discussions that we've had with even University of Colombo, we would be able to formalize these structures and come in with a program by which such kind of scholarships get encouraged. It may be small steps, but trust me, no steps will be backwards. That's the important part, and they will never stop. So that's something that certainly I would like to assure you. I would, in her absence, I would like to communicate a very big thank you to Professor Priyani Paranagama. She has been a great source of inspiration. She may be chair of chemistry in University of Kelaniya, but her insightful queries have always propelled a lot of work for us to do. And I thank her today for her presentation, as I thank Professor SKMK Herapathinia. Madam, you have thrown great light on the safety studies, and I hope such and like safety studies, we can hasten the pace and move them from not only chronic to a different set of minerals also that we can bring about. It's something to be looked forward to. Dr. Shubhamangala Acharya, a very leading practitioner, even while sitting here and waiting for her turn, as well as after her turn, she has been continuously talking. I'm sure these are patients. These are, dispensary people from her colleagues from her dispensary who are continuously calling. And, she's been brief, but, she's at it. And Madam, thank you again for sparing your time and coming across and sharing your experiences. I think it was very insightful and a very different manner in which a practitioner takes it to heart and really gets involved with even the patient's psychosis before the cure is really brought on. Thank you, Madam. Dr. Yogesh Kulkarni, you have always been a good scientific friend, I must say, and I hope we have also enthused you sufficiently, because exactly as Chairman Sir said, this is just the beginning. And what we have done is only conceptually imagined pilots and taken them to some particular state. The real test and the real science is something that follows. Dr. Priyadarshini Gunaratne, Madam, in your presentation, after all the theories, you brought in the various pictures from your practice. And that was just an amazing, amazing sense of satisfaction in the manner in which you have employed basic principle. And most importantly, the kalpana of Rugnahit, how you have upheld it at all points of time. I cannot even imagine the gratitude, the blessings that you have received from all those patients whose limbs you have managed to save. Thank you once again for taking time and coming across. I did tell my own colleague, Dr. Mukesh Chawda, to cut 5 minutes in his entire speech. He did take 15 minutes more. But you could see the scientist in him. You could see the enthusiasm in him, in which he wanted to present the kind of scientific discourse that is today on in our company. And yes, he has been a core architect in all our research work. And I'm sure, like you have received this particular book, which is a report card of the last 5 years of work, periodically you will be hearing much more from all of us, because all of those institutes that we have been able to tap into and activate, there are various projects that are going on right now. And we encourage even MD students to speak to the company. One of the things that we don't like only to do with an MD student, I'll be very blunt, most of the MD students call us for the medicines. What we want is actually the MD students to come and manufacture the medicines themselves, do the quality control themselves, do the standardization themselves, and then take the medicines out for whatever trials that they want to do. This is the kind of engagement we would like to have with scholars. We are not out here only to give a discount or only to provide the medicines. An MD thesis to us is a new leaf of knowledge that's added to this great science called Ayurveda. And that has to be done to that level of seriousness. It cannot be just a postgraduate degree that is just achieved by a particular thesis paper. And I hope we can meet students who kind of share this entire thought process. I would also like to thank the audience, leading practitioners again from Colombo and all around, students, scholars. I can see there are a lot of researchers also that have come from other disciplines. There are surgeons, of course. There are consultants. I can see a lot of faculty also that has taken time to come. As we have seen, all the provincial commissioners also have taken time to travel. It must have taken you, sirs and madam, almost 4 and 6 hours to come, but I really do thank you for taking the time and coming to this entire Sambhashan. And, we are going to hopefully have it every year, and we will go on adding a lot more feature to it. And certainly after today and the delay that has taken place, we would break it up into sessions and not just make it 1 or 2 keynote speakers, by which it's a more comfortable kind of arrangement and a layout for the entire conference. And probably we get more scholars to come and share their experience. With these words, I would end my vote of thanks and thank each one of you from everyone in Sri Dhootapapeshwar for sparing your time and giving us the honor of your gracious presence here. Thank you.
Thank you very much, sir, Mr. Puranik, the Managing Director and the Chairman of Sri Dhootapapeshwar Limited. Standing along 150 years is an onerous task. Some words quoting from Mr. Puranik's speech: yes, yet small steps, but confident, not taking back. That might be the success story of having such a tremendous journey and establishing their practice here in Sri Lanka more than a decade, I think nearly 1.5 a decade. Giving the Ayurveda practitioners what is expected to treat patients in a more efficient manner. Alpa matropayogitvat, as I mentioned previously, with tiny doses without giving a bunch of medication. Today we have realized the importance of Rasashastra to a greater extent. Thank you very much, ladies and gentlemen, for being here. And once again, a very big thank you for Sri Dhootapapeshwar Limited for giving this opportunity for practitioners, consultants, and specialists, even the students. They got together to learn more and to engage in this practice. To a certain extent, if we were doing something here in Sri Lanka that will boost the Pithora to exceed more and succeed more in the future as well. We're expecting you the next year as well, this type of conference, this type of knowledge sharing experience that itself Rasashastra has been mentioning in its ordinary text. So we need to share knowledge, we need to share our expertise, and we need to have our experience with the patient. Not to treat when it comes to even prevent at the initial stage. Let's communicate this message to the entire country, and yet with small steps, but yet confidently, let's drive out the success. Thank you very much, and see you at the 7th episode soon here in Sri Lanka. Thank you very much, Sri Dhootapapeshwar Limited, participants, esteemed intellectuals. Thank you very much. Have a pleasant day. And a gentle reminder for all the speakers who are present here, we would request you to come in front, take a group photograph, and after that, your refreshments are ready outside. Thank you.